There are roughly 8 billion of us walking around on this planet right now. We eat, we sleep, we breathe, we bleed. And when we bleed, almost all of us carry one of just 4 basic blood types: A, B, AB, or O. Maybe you know yours. Maybe you had it written on a card at some point, or a doctor mentioned it once, or you saw it on a form before surgery. If you do know it, hold that in the back of your mind for a moment, because here is the question that quietly sits underneath all of medicine, underneath evolutionary biology, underneath thousands of years of human history. Why? Why do blood types exist at all? We are one species. We share something like 99.9% of our DNA with every other person on Earth. We came from the same small group of ancestors in Africa. We went through the same bottlenecks, the same ice ages, the same migrations. And yet, right there on the surface of your red blood cells, you carry a molecular signature that is genuinely different from the person sitting next to you. Not just slightly different. Different in a way that if a doctor gives you the wrong blood in a transfusion, your immune system will treat it like an invasion and try to destroy it. Different in a way that shaped which diseases your ancestors survived and which ones killed them. Different in a way that has been carried forward across hundreds of thousands of years of human evolution and is still sitting inside your veins right now. That is not a small thing. That is one of the stranger facts about being human, and most of us walk around never thinking about it for a single second. If you are listening to this to wind down, that's exactly why these episodes exist. So get comfortable. There is no rush here, and if you want to tell me where in the world you are and what time it is where you are, drop that in the comments. It is one of my favorite things. I open the comments, and it is Mumbai at midnight, Melbourne at 3 o'clock in the morning, somewhere in Norway at breakfast time. All of you all at once listening to the same question about blood. I find that genuinely remarkable every time. And if you want to support what this channel does without seeing any ads and without any background music getting in the way, the best thing you can do is follow on Spotify. No ads, no music, just the voice and the story. The link is in the description below. Now, back to the blood. The mystery of blood types is actually 3 mysteries stacked on top of each other, and this episode is going to work through all 3 of them. The first is a medical mystery. For most of human history, if someone was bleeding to death, there was almost nothing a doctor could do. And then, about 120 years ago, a scientist in Vienna figured out that human blood came in different types, and that certain combinations were deadly and others were safe. That discovery saved millions of lives and completely transformed surgery and emergency medicine. But it also raised a deeper question that the medical community could not answer at the time. Why are the types different? Why does this variation exist? That leads to the second mystery, which is evolutionary. Blood type differences are not random noise. They are ancient. The blood group system is so old that it actually predates our species entirely. Chimpanzees have blood types. Gorillas have blood types. Some of the same molecular differences we carry in our veins today were already present in primate populations millions of years before modern humans existed. And that tells you something important. These differences have been maintained across enormous stretches of evolutionary time, which almost certainly means they were doing something useful. Something that helped certain individuals survive things that killed others. Disease is the most likely answer. Specific pathogens, things like malaria and cholera and smallpox, appear to interact with blood type in ways that gave some people a survival advantage. And over thousands of generations, those advantages quietly shape the distribution of blood types across different populations around the world. In some regions, type O dominates. In others, type E is far more common. That uneven map is not an accident. It is a record of which diseases swept through which places and who survived them. And then there is the third layer, which is the history of how humans have tried to understand all of this, because the science of blood types did not arrive cleanly and all at once. It came in pieces through mistakes and accidents and slow realizations. Doctors tried blood transfusions for centuries before they understood why some patients died and others lived. The Rh factor, the positive or negative part of your blood type, was only discovered in 1940, and its implications for pregnancy were not fully understood until even later. And running alongside the science, winding through it like a shadow, there is a whole other history of folklore and myth and cultural meaning that people attach to blood types, especially in Japan, where your blood type is still sometimes used to guess your personality, the way other cultures use astrology. That part of the story is fascinating in its own right, because it tells you something about how humans take a biological fact and make it into a story about who we are. So that is where we are going. Evolution, disease, and the history of medicine, threaded through with ancient DNA and population genetics, and the occasional moment where a scientist discovered something so strange, it took years for anyone else to believe it. But let's start with a simple version first, because before we can talk about why blood types exist, it helps to understand what a blood type actually is, what is physically happening on the surface of that red blood cell, what your immune system is reacting to, and why getting the wrong type in a transfusion is not just a bad outcome, but a biological emergency. 'Cause here's the thing. When you look closely at what a blood type is at the molecular level, the whole mystery gets even stranger. These are not complicated structures. They are not deeply fundamental parts of how blood works. They're relatively simple molecules sitting on the outside of a cell like little flags. And for a long time, nobody could figure out why the body cared about them so intensely, why the immune system had learned to treat an unfamiliar blood type as a threat it had never even encountered before. That does not make obvious sense, and the explanation, when it finally came together across decades of research, turns out to involve the immune system. The history of infection and evolutionary pressures, so old they reach back before our species, had a name for itself. Somewhere in your body right now, on the surface of every red blood cell you have, there are molecules that mark you, that say something about where your ancestors came from, what diseases they survived, what evolutionary pressures shaped the people whose blood eventually became yours. You are carrying ancient biological history inside you, without knowing it. Most of us never stop to think about it, but the story it tells once you start pulling on the thread turns out to be one of the longest and strangest in all of human biology. Let's start pulling. Your red blood cells are small, impossibly small. A single drop of blood contains somewhere around 5 million of them, and each one lives for roughly 120 days before your body breaks it down and makes a new one. They spend their entire short lives doing one thing: carrying oxygen from your lungs to your tissues and carbon dioxide back again. Simple, functional, unremarkable at first glance, but the surface of each one of those cells is covered in molecules. Proteins and sugars attached to the outer membrane like decorations on a very crowded surface, and some of those molecules, specific ones, are what we call antigens. An antigen is essentially anything the immune system can recognize and respond to. It is a shape, a molecular signature that your immune system learns to read. And the particular antigen sitting on the surface of your red blood cells are what determine your blood type. In the system, the one most people know, there are two antigens in play, antigen A and antigen B. If your cells carry antigen A, you are type A. If they carry antigen B, you are type B. If they carry both, you are type AB. And if they carry neither, you are type O. That is the whole system at its core. 4 outcomes from 2 possible antigens. Now, here is where it gets important. Your immune system does not just passively notice what is on your own cells. It also produces antibodies against antigens it does not recognize. And those antibodies are where the danger comes in. If you are type A, your immune system carries antibodies against antigen B, the one you do not have. If you are type B, you carry antibodies against antigen A. Type O carries antibodies against both. Type A carries neither, because it recognizes both antigens as self and does not need to attack them. This is why mismatched blood in a transfusion is not just medically inconvenient. It is a biological emergency. Your antibodies recognize the foreign antigens on the incoming red blood cells, bind to them, and trigger a cascade. These cells begin to clump together, a process called agglutination. Then they rupture. The immune system escalates. You can go into shock. Your kidneys can fail. People have died from it. Not because the wrong blood was toxic in any direct sense, but because the immune system treated it the way it treats an infection with full force, and the collateral damage was catastrophic. The Rh system works by a different set of rules. The molecule involved here is a protein called the Rh factor, most commonly the D antigen, and it operates independently of entirely. You either have it Rh positive or you do not Rh negative. The key difference is that Rh antibodies do not appear spontaneously the way antibodies do. Your body only produces anti-Rh antibodies if it is actually exposed to Rh positive blood, when it does not have the antigen itself. That first exposure may cause little obvious harm, but the immune system remembers, and a second exposure can trigger a severe reaction. This is especially significant in pregnancy, where an Rh negative mother carrying an Rh positive baby can develop antibodies that cross the placenta in subsequent pregnancies. And we'll get into exactly why that matters in more detail later. The point is that and Rh are not the same system, do not involve the same molecules, and carry different kinds of risk. They just happen to get reported together on the same blood type card. What blood types are not, and it is worth saying this clearly, is a window into your personality. The idea that blood type predicts whether you are creative, or disciplined, or difficult to get, along with, has no biological basis. Red blood cell antigens do not cross into the brain. They do not shape cognition or temperament. That is folklore, and we will look at how that folklore developed, and why it took hold in certain cultures. But it is not biology. What blood types are biologically is an example of what geneticists call a polymorphism, a trait that exists in multiple stable versions within a species at the same time. Polymorphisms are interesting precisely because they persist. Natural selection tends to push populations toward whatever version of a trait is most advantageous and eliminate the others. When a species maintains multiple versions across long spans of time, it usually means the different versions are doing something, surviving different pressures, conferring different advantages in different contexts, and that is the clue buried inside the existence of blood types. We can often make educated proposals about what those pressures were. Pinning them down exactly with certainty turns out to be much harder than it sounds. Once you know all of that, the neat categories, the antibodies, the agglutination, it is very easy to forget that for most of medical history, no one had any idea any of this existed. Blood was a symbol, a humoral fluid, a kind of essence. It was not a precisely defined tissue with invisible molecular identities. It was life itself in liquid form, which is why once people started trying to move it from one body into another, the results looked almost like a cruel lottery. The very first transfusion experiments in the 17th century were not even human to human. They were driven by curiosity and a kind of hopeful speculation. What if you could calm a disturbed mind by giving it the blood of a gentle animal? What if the vigor of a lamb could repair a failing body? Physicians and natural philosophers in France and England bled dogs and sheep and tried, pumping their blood into other animals and eventually into humans. Sometimes the subject survived and appeared to recover from whatever condition they had. Sometimes they became violently ill or died. No one could predict which way it would go because no one knew they were playing with immune systems. They were looking at a dark stage and only seeing what the candlelight of clinical observation could reveal: a pulse, a skin tone, a breathing pattern. If a patient woke up feeling better, the transfusion was praised. If they died, the transfusion was blamed. There was no way to separate in any rigorous sense what the blood itself was doing from what the underlying illness, or the crude equipment, or simple bad luck was doing. After a few spectacular failures, European authorities grew uneasy. Transfusion trials were restricted or banned. For almost 2 centuries, the idea mostly went quiet. Physicians kept bleeding patients the old-fashioned way, taking blood out rather than putting it in, because that fit their theories about balancing humors. If someone lost a dangerous amount of blood in childbirth or in an accident, there was not much anyone could do. You hoped the bleeding stopped. You waited. Sometimes the patient survived. Often they did not. The problem was not that earlier doctors were careless. Many of them were meticulous observers. They kept careful notes. They agonized over every death, but all they could see were patterns on the surface. Some patients seemed to tolerate transfusions from certain donors better than others. Animal blood usually went badly, though not always immediately. Even human blood gave mixed results. Two desperately anemic patients, same hospital, same doctor, apparently similar conditions. One received blood and recovered. The other received blood and went into convulsions, passed dark urine, developed fever and pain, and died within hours. To us, with our charts of AB, and O, this looks obvious in retrospect. Donor and recipient were probably incompatible. The recipient's antibodies attacked the transfused cells, clogged the kidneys, triggered shock. But to a 19th-century physician, there was no such framework. They might note that the blood seemed to disagree with the patient and move on, unsure whether the problem was the donor, the disease, the timing, or some intangible quality of the patient's constitution. And yet, underneath the confusion, patterns were slowly emerging. Surgeons operating before the era of modern transfusion knew that some families contained good givers, people whose blood seemed more likely to help than harm, though no one could say why. Midwives and obstetricians saw the same. A woman hemorrhaging after childbirth might be saved by blood from her sister, but not from her husband. The outcome seemed almost superstitious, as if compatibility were a matter of fate or character. Clinical observation was ahead of understanding. Doctors could describe what was happening in excruciating detail. They could not explain it. They recorded temperatures spiking after transfusion, urine turning red or brown, patients complaining of heat and anxiety, of crushing pain in the back and chest. They saw that these reactions tended to occur quickly, often within minutes. They knew that smaller volumes of blood were sometimes tolerated when larger volumes were not. But the invisible confrontation between antibodies and antigens, the precise reason why one person's blood flowed harmlessly in another's veins, while a different pairing was lethal, lay far beyond the reach of the microscopes and chemistry of the time. The stakes were especially clear in three settings: war, childbirth, and surgery. On battlefields, surgeons watched young, otherwise healthy soldiers bleed to death, from injuries that might have been survivable if their circulating volume could be restored. During childbirth, obstetricians saw the same thing in civilian life. A complication, placenta previa, uterine, a tear could turn a routine delivery into a crisis within minutes. In the operating theater, as surgical techniques improved and more ambitious operations became possible, the limiting factor was often blood loss. You could remove a tumor, repair a wound, or attempt to fix an aneurysm, but if the patient lost too much blood on the table, all your skill meant nothing. These were not abstract problems. They were everyday tragedies. Physicians knew with overwhelming clarity that if they could safely give blood, lives could be saved that were otherwise doomed. The urgency was not theoretical. It was written in mortality statistics and in the memory of every doctor who had watched a patient fade away, fully conscious, simply because their heart had nothing left to pump. So transfusion kept coming back. Each new generation of doctors revisited it with slightly better tools. By the late 19th century, they could prevent clotting with simple techniques. They could attempt to match donors and recipients by Crudder measures, like observing whether small samples of blood mixed in a glass tube seemed to form clumps without fully understanding what that clumping meant. Some surgeons developed elaborate methods of direct transfusion, stitching donor and recipient blood vessels together with delicate tubing, hoping that moving blood directly from one person into another might be safer than collecting it in a container. Results improved a little, but they were still maddeningly inconsistent. Some hospitals reported encouraging survival rates, others saw devastating reactions and abandoned the practice again. There was no shared language to describe compatibility, no categories, no way to predict in advance which pairings would be safe. Every transfusion was essentially a gamble, a high-stakes experiment played out in the veins of someone already dangerously ill. When blood groups were finally identified and their clinical significance recognized in the early 20th century, It did not feel like a small refinement. It felt like someone had quietly turned on the lights in a room where medicine had been stumbling in the dark for decades. The idea that blood could come in distinct types, defined by specific inheritable traits on red blood cells, suddenly made sense of that long history of chaotic results. The good givers and bad reactions were no longer mysterious quirks. They were consequences of antigen patterns and antibodies that could be measured, named, and crucially avoided. Transfusion, once a desperate last resort, became something that could be planned. Instead of hoping that donor and recipient would be lucky, doctors could test their blood in advance, see whether it reacted, and match them according to known rules. War medicine changed, obstetrics changed, surgery changed, mortality from hemorrhage began to drop, and the same molecular differences that had quietly divided humanity into A, B, AB, and O for hundreds of thousands of years suddenly acquired a very immediate meaning at the bedside. Who could safely receive whose blood? But acceptance was not instant. Medicine rarely shifts all at once. Some clinicians were skeptical. Others lacked the resources or training to implement typing and cross-matching consistently. Different hospitals adopted the new methods at different speeds. For years, there was a patchwork of practice. Some institutions meticulously typed every patient. Others still relied on rough visual tests or nothing at all. Each transfusion pushed the profession one way or another. A successful, well-matched transfusion encouraged adoption. A fatal mismatch, in a place that had ignored the new knowledge, was a grim reminder of what was at stake. Over time, the evidence accumulated. Typed and cross-matched transfusions produced dramatically fewer reactions. Blood banks began to form. Standard protocols spread through military medicine, then into civilian hospitals. What had once been an uncertain, almost magical intervention became a routine part of care grounded in reproducible science. And underneath that transformation, the same question that opened this story remained. Why do these blood types exist in the first place? The discovery of, and later Rh, turned transfusion from a gamble into a planned procedure. But it also revealed just how deeply structured our blood really is, and it raised a new mystery. If these tiny differences could mean the difference between life and death on the operating table, what have they been doing across the long sweep of human evolution? The person who finally put names to those tiny differences was an Austrian pathologist named Karl Landsteiner. He was not trying to solve all of transfusion medicine at once. He was doing what many great scientists do in hindsight, asking a very simple question extremely carefully. He wanted to know why, under the microscope, blood from different people sometimes behaved differently when you mixed it. At the time, it was known that if you took a drop of blood and let it settle, you could separate the red cells from the clear fluid around them, the serum. The serum was where antibodies lived, though the word antibody was still a relatively new idea. Landsteiner suspected that whatever was causing some transfusions to go wrong was hiding in that interaction between red cells and serum. So, he started mixing quietly, patiently, methodically. He took small samples of blood from his laboratory colleagues, spun them down, separated red cells from serum, labeled them, and began combining them in every possible pairing. Serum from person 1, with cells from person 2, serum from person 1, with cells from person 3, serum from person 2, with cells from person 1, and so on. He kept track of what happened. Most of the time when serum from one person met the red cells of another, nothing much happened. The cells stayed evenly suspended, drifting like tiny discs in the liquid. But sometimes, something striking occurred. The red cells would suddenly clump together, forming little clusters. They did not dissolve, they stuck. To a modern hematologist, this is simple agglutination. To Landsteiner, it was a clue that some bloods were fundamentally incompatible in a way you could see. He noticed that these clumping reactions did not happen randomly. The same combinations of donor and recipient kept producing the same outcome. Serum from one particular colleague would always clump the cells of some people and never clump the cells of others. Another colleague's serum had a different pattern. When Landsteiner arranged the results in a table, there was a structure there like a code emerging from the noise. From that pattern in 1900 and 1901, he proposed the first three blood groups, A, B, and C, not yet A, B, AB, and O. The group would later be renamed O for Owen, the German word for without, because those red cells seemed to lack the two main markers he had found. Shortly after, his team and others realized that some people's red cells carried both markers. That fourth group became AB. What Landsteiner had stumbled onto was both simple and profound. Some red cells carry one kind of sugar-decorated protein on their surface. Those were type A. Some carry a different kind. Those were type B. Some carry both AB. Some carry neither O. These surface markers became known as antigens A and B. In the serum, meanwhile, live the antibodies. They were like tiny floating centuries. In most people, the pattern was again orderly. If your red cells had A antigens, your serum contained antibodies that reacted against B. If your red cells had B antigens, your serum reacted against A. If you had neither antigen, type O, your serum carried both anti-A and anti-B antibodies. If you had both antigens, type AB, your serum carried neither. This is why the clumps appeared in Steiner's bowls and tubes. When a person's serum met red cells carrying an antigen that person didn't have and had antibodies against the antibodies latched onto the foreign antigen, each antibody can bind more than one cell, bridging them together. Many bridges at once, and the cells knit into visible clumps. Agglutination under the microscope. It looked almost gentle, like snowflakes gathering into drifts. In a living body in the wrong veins, it could be lethal. The rules that students memorize today can receive from A and O, B from B and O, AB from everyone, O, only from O, are just a tidy way of summarizing those simple antibody-antigen relationships. If you give someone blood whose antigens their serum is ready to attack, you're setting up that clumping reaction inside their circulation. The transfused cells break apart, release their contents, clog tiny vessels, and strain the kidneys. Shock follows. The patient can die. If, on the other hand, you give someone blood whose cells carry no antigens that their antibodies can recognize, the transfused cells slip into the bloodstream quietly, like guests who have been properly introduced. Type O red cells, in the sense, are in that position for most recipients. They carry neither A nor B antigens. So even if the recipient has anti-A or anti-B, or both, there is nothing for those antibodies to grab onto on the donated cells. This is why O so quickly gained a reputation as the universal donor. In the emergency setting, when there is no time to carefully type and cross-match, type O red cells can often be given more safely than unknown AB or AB. The soldiers on the front lines of the world wars, the victims of accidents before modern blood banking systems became widespread. Many of them survived because someone had a unit of O on hand. But universal here is a careful, limited word. It really refers to red cells alone, and even then, it's only in the system. Blood has many other antigen systems like Rh, Kell, Duffy, and more. Serum contains not only antibodies, but potentially others as well, especially in people who have been previously transfused or pregnant. When whole blood is given, the donor's serum with its antibodies matters too. A type O person, for instance, has both anti-A and anti-B in their serum. If you transfuse large amounts of their whole blood into a type A recipient, those donor antibodies could attack the recipient's own red cells. This is why modern transfusion medicine is precise. In routine practice, people receive packed red cells, plasma, or platelets separately. Each component is matched not just for, but when possible, for several other antigens. The old idea of a single kind of blood that anyone can safely receive has given way to a more nuanced picture. Type O red cells are widely useful. Type AB plasma, which contains no anti-A or anti-B antibodies, is in some ways the universal plasma, but every pairing is still evaluated in context. Landsteiner's original work, however, did something perhaps even more important than providing practical rules. It revealed that these types were not rare oddities. When other researchers began to test large numbers of people, they found that all 4 groups were common. Their frequencies varied, but no population was entirely one type. In some parts of Europe, A was particularly common. In others, B was more frequent. Among many indigenous groups in the Americas, O dominated. In parts of Asia, the distribution of AM looked different again. It was as if Landsteiner had drawn the first simple map, and then more data points filled in the coastlines and mountains. Each community, each region had its own pattern of A, B, AB, and O. Those patterns were not random. They reflected deep histories of ancestry, migration, isolation, and mixing. Later, as anthropologists and geneticists joined the story, frequencies became one of the tools for tracing human population movements alongside languages, artifacts, and eventually DNA sequences. But the map raised an evolutionary puzzle. In a small, isolated population, it is easy for one genetic variant to drift to high frequency or even to become fixed just by chance. Over long stretches of time, you might expect one blood type to become overwhelmingly common or other types to fade away, especially if they carried any disadvantages. Yet around the world, all 3 main alleles that create the system, A, B, and O, have persisted for hundreds of thousands, perhaps millions of years. No single one has quietly erased the others. That persistence is the real mystery Steiner uncovered, even if he did not frame it in those terms. What has kept A, B, and O all in circulation, in different proportions, in so many human groups, and in other primates as well? Why should natural selection tolerate, and in some places even favor such variation, instead of pushing us all toward a single optimal type? The answer, as far as we can tell, does not lie in transfusion at all. For almost all of evolutionary history, no one was swapping blood between individuals. Whatever forces were shaping frequencies had to be acting in the ordinary business of life. Infection, reproduction, diet, the constant, quiet negotiation between host and environment. Once Landsteiner had shown that blood types were real, inheritable traits, the next step was to ask what they were for, if anything, beyond confusing early transfusionists. The clues to that deeper story begin with another discovery, made only a few decades later, on the surface of the very same red cells. The next clues came from a different set of tiny markers on the red cell surface discovered in the 1940s. By then, scientists were used to the idea that antigens mattered for transfusion, but patients were still having mysterious reactions, even when the match was perfect. Something else was hiding in the membrane of those cells, something Landsteiner had missed in his original work. This new system came to be called Rh, after the rhesus, macaque monkeys, used in some of the early experiments. The name is a historical accident. The most important antigen in this system, the one people usually mean when they say Rh positive or Rh negative, is a particular protein called D. If your red cells have the D antigen, you're Rh positive. If they don't, you're Rh negative. This Rh-D protein is not a decorative sugar like A and B. It's a complex structure that sits in the red cell membrane and seems to be involved in how the cell handles certain ions like ammonium. All modern humans have the Rh gene cluster, but different versions of the genes, and sometimes large deletions, determine whether the D antigen is actually present on the surface. From a transfusion perspective, the rule is simple enough. An Rh negative person has no D antigen. If they receive Rh positive blood, their immune system can recognize that D protein as foreign, and over time make antibodies against it. Once they have those antibodies, anti-D, any future encounter with depositive red cells can trigger a rapid and sometimes dangerous immune reaction. So in modern blood banks you match people for Rh at a minimum. A person who is O negative, for example, lacks AB and D antigens. Their red cells can be given in emergencies to a wide range of recipients. But for evolution, and for the history of ordinary lives, the Rh story turned out to be most important, not in transfusion rooms, but in pregnancy. Unlike antibodies, which most people develop very early in life, probably through exposure to gut bacteria that mimic A and B antigens, anti-D antibodies usually do not appear unless the immune system has been clearly exposed to the D antigen. That exposure often happens during childbirth. Imagine a woman who is Rh negative. Her red cells carry no D antigen. Now imagine that her partner is Rh positive and that the fetus they conceive inherits the D antigen from the father. The baby growing in her uterus is Rh positive. For most of the pregnancy, mother and fetus do not mix large amounts of blood. Nutrients, gases, and waste cross the placenta, but red cells mostly stay on their own sides. Still, pregnancy and especially birth are not perfectly sealed processes. Tiny leaks happen during delivery, or if there is trauma, or sometimes even in late pregnancy. A small number of fetal red cells can cross into the mother's circulation. To her immune system, those D-bearing cells are unfamiliar. They carry a protein her own blood does not. The first time this happens, there is usually no immediate catastrophe. Instead, her immune system does something that is both ordinary and, in this context, quietly dangerous. It learns. It processes the foreign D antigen, activates specific lymphocytes, and produces a small amount of anti-D antibodies. More importantly, it creates memory cells, immune cells primed to respond much more vigorously if they ever see D again. This process is called sensitization. It is the same basic principle that makes vaccines work. The first exposure creates a memory so that future exposures are met with a rapid, strong response. In infections, that is protective. In Rh incompatibility, it sets a trap for future pregnancies. Now picture a second pregnancy. The same Rh negative mother conceives another Rh positive fetus. For many months, things may seem outwardly normal. Inside, however, the mother's immune system is different from what it was the first time. It remembers D. Very small amounts of fetal blood crossing the placenta are enough to wake those memory cells. They begin producing anti-D antibodies in large quantities. Antibodies are small. They can cross the placenta easily. When they do, they meet the fetal red cells in their own circulation. And this time, unlike in Landsteiner's laboratory bowls, the clumping is only part of the story. Anti-bide antibodies coat the fetal red cells and mark them for destruction in the fetal liver and spleen. The baby's body tries to compensate, making more red cells, even pushing immature cells into the bloodstream, but the balance can tip. The result is hemolytic disease of the newborn, sometimes called erythroplastosis for Talus in older texts. Hemolytic because red cells are being broken apart. Erythroblastosis because many immature red cell precursors appear in the blood as the body desperately tries to keep up. In severe cases, the fetus can develop profound anemia, swelling, heart failure, and without intervention, die before or soon after birth. It is an immune memory problem stretched across pregnancies. The first rosative child of an Rh negative mother is often unaffected. It is their existence and the fetal cells that escape during their birth that primes the mother's immune system. It is the second, third, or later Rh-positive child that bears the brunt of that memory. Before the mechanisms were understood, this pattern showed up in heartbreaking statistics. In some families, first babies would be healthy, while younger siblings were still born, or died, in the first days of life with jaundice and anemia. In certain populations where Rh-negative women were common, and Rh-positive men equally common, this was a recurrent, quiet disaster. The turning point was not a new evolutionary change, but a new medical strategy. Prevent sensitization in the first place. Once researchers established that hemolytic disease in many of these cases was caused by maternal anti-D antibodies, the logic became clear. If you could stop the mother's immune system from ever learning D as foreign, you could protect all her future pregnancies. The tool they developed for this was Rho immune globulin, often known by trade names, but conceptually very simple. It is purified anti-D antibodies taken from donors administered to an Rh-negative mother at specific times usually late in pregnancy and shortly after delivery of an Rh-positive child. These passive antibodies quietly mop up any fetal Rh-positive red cells that cross into her circulation before her own immune system has a chance to react to them. They bind and help clear the cell so quickly that the mother's lymphocytes never see enough d-antigen in the right context to become sensitized. No memory cells form. The mother remains immunologically naive to D, even though she has carried an Rh-positive baby. This is one of the rare cases where medicine deliberately prevents the immune system from doing what it naturally wants to do, or form a lasting memory. The effect on outcomes has been dramatic. In countries where routine Rh screening and RO prophylaxis became standard in the late 20th century, severe RF-mediated hemolytic disease of the newborn, once a major cause of infant death, became rare. When it does occur now, it is often in settings where screening and prophylaxis are not yet universal, or in complex cases involving other blood group antigens. All of this might make Rh-negative status sound like an inherently dangerous or unusual condition, but it is neither universal nor vanishingly rare. Its frequency varies with ancestry. In parts of Europe, especially the Northwest, Rh-negative individuals can make up around 15% of the population. In much of East Asia and Africa, the proportion is much lower. Among some indigenous American groups, it is extremely rare. These differences almost certainly reflect population history, founder effects, drift, and path selection, rather than any mystical property. Rh-negative is simply one of the common variants that emerged and persisted as human groups, moved, split, and rejoined. In small ancestral populations, chance events could increase or decrease its frequency. If there were any advantage or disadvantage associated with Rh status in particular environments, perhaps through subtle effects on infection risk or reproduction, those pressures would have acted slowly over many generations. The modern pattern is the outcome of that long, complicated history, not the fingerprint of a single dramatic event. What the Rh story does underline is that blood types matter in more domains than we might first assume. ABO differences influence transfusion compatibility and may shape how certain infections interact with the body. Rh differences can turn one pregnancy into the immunological prelude to the next. The markers on our red cells are not just passive labels. They participate in the ongoing dialogue between immune system reproduction and survival. As we look more closely at these systems, a broader pattern emerges. Variation in blood groups has been preserved not just because of accidents of history, but because in different times and places, different variants can be useful, or at least not harmful enough to disappear. To understand why A, B, O, and the R types have all lasted so long, we have to look beyond transfusion rooms and maternity wards out into the wider environment where the fiercest, most ancient pressures on red blood cells still come from. Parasites, particularly the ones that live in blood. So far, we've been talking as if blood types are just there, like fixed labels stamped onto humanity. But in evolution, just there is rarely the whole story. Traits that cost the body energy that can trigger fatal transfusion reactions, or pregnancy. Complications don't generally stick around for hundreds of thousands of years by accident. If there were one obviously best blood type in every environment, natural selection would have pushed us all in that direction long ago. Yet when researchers look at human populations, they keep seeing the same thing. Multiple blood group variants at substantial frequencies in almost every region that's Rh. Not just for and Rh, but for dozens of other systems too. In evolutionary biology, that kind of persistent variation has a name. It's called a polymorphism, and the fact that it persists generation after generation begs a deeper question. Why hasn't evolution simply picked a winner and erased the rest? One of the main answers biologists have arrived at is something called balancing selection. Instead of a single best variant sweeping to dominance, the environment and the rules of survival work together in a way that keeps several variants in play. No single type is consistently superior in all circumstances. Sometimes one version does better, sometimes another, and over long stretches of time, The tug-of-war can hold multiple types in a kind of dynamic equilibrium. There are a few different ways balancing selection can work. One of the most intuitive is known as frequency-dependent selection. In this scenario, the advantage of a particular trait doesn't just depend on what the environment is like. It also depends on how common that trait is in the population. Imagine for a moment that you're not a human, but a pathogen, a virus, a bacterium, a parasite trying to spread through a community. You encounter red blood cells with certain surface patterns, again and again. Over generations, you adapt to those patterns. Your attachment proteins shape themselves by trial and error to match the most common antigens, the ones you see most often. In that setting, hosts with the common blood type become easier targets simply because you'd spent more evolutionary time solving the problem of how to infect them. Now, picture a person whose red cells carry a less common antigen pattern, some variant you rarely meet. Suddenly, your finely tuned machinery doesn't work quite as well. Maybe you can still infect them, but you do it less efficiently. Maybe you can't attach at all. To that rare host, your specialization becomes their advantage. They are, at least for a while, slightly harder to exploit. But the story doesn't stop there, because their advantage is tied to being rare. If that blood type begins to spread and becomes more common, pathogens will start seeing it more frequently. Over time, they adapt to it too. The advantage erodes. Being common becomes a liability. Being rare becomes once again a refuge. The result is a moving balance point where no single type ever quite takes over, because as it rises, the forces that push it back down get stronger. There are classic examples of frequency-dependent selection in nature. Color morphs in fish that sneak past guards, left and right-mouthed predators that attack from different sides, floral variants that trick pollinators. But something similar can plausibly happen with immune system markers and blood group antigens. Pathogens and hosts are locked in an arms race. Anytime one side gains a stable, predictable pattern, the other side can exploit it. Diversity from the host's perspective is a moving target. A related mechanism that can help maintain polymorphisms is heterozygote advantage. Instead of rare individuals being favored simply because they are rare, this is a case where individuals who carry two different versions of a gene do better than individuals who carry two copies of the same version. The most famous example does not involve blood types at all. In regions where malaria has been common for thousands of years, people who carry one copy of the sickle cell mutation and one normal copy of the hemoglobin gene often have some protection against severe malaria without developing full sickle cell disease. In that context, being a mixed genotype is better than either extreme. Natural selection, in effect, keeps the mutation around because in single copy it is useful with blood groups. The evidence is less clear-cut, but the principle is similar. Many blood group genes come in multiple functional versions. An individual might be A from one parent and O from the other, or carry one Rh variant that produces the D antigen and another that alters it. If certain combinations of these alleles provide broader protection against a range of pathogens than either version alone, then natural selection can favor the mixed heterozygous state. Over time, that can help preserve both variants in the population, even if neither would be as successful on its own. These are adaptive explanations, stories about how selection can actively maintain diversity. Because diversity itself is useful, but evolution has another side to it, one that operates without regard to usefulness at all. It is the realm of chance. In small populations, genetic drift can be a powerful force. Drift is simply the random fluctuation in gene frequencies that occurs because only some individuals happen by luck to leave descendants. Imagine a tiny group of humans crossing a land bridge or an ice age valley. If just by chance more of them have a particular blood type, that type will be overrepresented in the next generation. Not because it helped them cross the valley better, just because those were the people who survived and reproduced over many generations, especially in scattered, bottlenecked populations. These random shifts can dramatically reshape the genetic landscape. A blood group AL that was once rare in the ancestral population can become common in a descendant group if it happened to be carried by the founders. Conversely, a once common AL can be lost entirely if by misfortune none of the people who carried it had children in a particular lineage. This is what geneticists mean by founder effects, the lasting imprint of those first few individuals who started a population in a new place. When we see that Rh negative is more frequent in parts of Europe than in East Asia, or that certain patterns cluster in particular indigenous American groups, we are looking not only at ancient battles with pathogens, but also at the ghosts of small migrating bands, of villages founded and lost, of lineages that expanded or dwindled for reasons that had little to do with blood. So when we ask why blood group polymorphisms persist at all, the answer is not a single clean story. It is a layered one. Some of the diversity we see today is almost certainly the product of balancing selection. Environments where no one blood type is consistently best, where pathogens constantly reshape the fitness landscape, and where being rare or being a heterozygote can offer real advantages. Some of it is history, drift, founder effects, bottlenecks and expansions that amplified certain variants and muted others without any conscious design. And sitting at the intersection of these forces are the pressures we can actually study most directly. Infectious diseases, parasites, bacteria, and viruses live and die on timescales much shorter than ours. They leave traces in our genomes in patterns of variation that stand out against the background noise of drift. When we look closely, we find hints that certain blood group alleles are associated with resistance or susceptibility to particular infections. We can see in the distribution of those alleles shadows of past epidemics and local disease, ecologies. Among all the pressures that could have shaped blood group diversity, diet, reproduction, climate, pathogens turn out to be some of the most powerful and testable. They attack the very cells that carry these antigens. They interact directly with the immune system that reads those molecular signatures. In the next part of this story, we'll follow them into the bloodstream to one of the most relentless and ancient enemies of human survival, and to a region of the world where your blood type could mean the difference between life and death. To understand why blood traits are such a favorite target of natural selection, it helps to start with what malaria actually is trying to do. The parasite that causes the most deadly form of human malaria, Plasmodium falciparum, has a very specific lifestyle. It doesn't just float around in the blood at random. After entering the body through a mosquito bite and spending a brief phase in the liver, it moves into red blood cells and turns them into tiny parasite factories. Red blood cells are tempting targets. They are abundant, simple, and lack nuclei. They don't present antigens to the immune system the way many other cells do. Once inside, the parasite can hide, reproduce, and then burst out in synchronized waves, infecting new cells. Those waves are what drive the fevers and chills that have haunted human populations for tens of thousands of years. But the parasite doesn't just sit quietly inside the cell. It remodels the red blood cell surface, sending its own proteins up to the membrane. These altered surfaces make infected cells sticky. They cling to the walls of tiny blood vessels in the brain, the placenta, and other organs. This sequestration keeps them from being filtered out by the spleen, but it also creates blockages, inflammation, and sometimes catastrophic damage. If you are a parasite, this is an elegant strategy. If you are a human, it is a disaster, and it creates an obvious arena for natural selection. Anything that changes the red blood cell surface, or the way those cells behave in capillaries, has the potential to influence how well the parasite can invade, hide, or stick. That is why so many of the clearest examples of recent human evolution involve red blood cell traits in malarial regions. But here we have to be careful. Not all of those traits have much to do with, or some of the best documented malaria-related variants are almost household names. The sickle cell mutation, which alters the structure of hemoglobin, is one. People who inherit 2 copies of the mutation often develop sickle cell disease, a serious and painful condition. But people who inherit just one copy in many African populations have a measurable reduction in the risk of severe malaria. Their red blood cells are just off enough, under low oxygen conditions, to interfere with the parasite's life cycle. Another example is the Duffy blood group, a different system from or Rh. In much of Western Central Africa, a particular version of the Duffy gene leads to red cells that don't express the Duffy antigen on their surface. One species of malaria parasite, Plasmodium vivax, uses that antigen as a doorway. In people with the Duffy negative type, that doorway is essentially gone, and infection becomes much harder. The very high frequency of Duffy negative in these regions looks like a textbook case of intense selection by malaria. There are others, glucose 6-phosphate dehydrogenase deficiency, and more subtle changes in red blood cell shape and metabolism. Collectively, they paint a clear picture. Malaria has been one of the dominant selective pressures on human blood physiology. Where does fit into this? The system changes the sugar structures displayed on the surfaces of red blood cells and some other tissues. Those sugar patterns, the A and B antigens, can be recognized by antibodies, and they can also be recognized by pathogens. It is reasonable to ask whether malaria parasites see and respond to those patterns. The evidence here is more nuanced than in the Duffy or sickle cell stories. Many studies, especially in parts of Africa and Asia, where falciparum malaria has been intense, have tried to see whether certain types are overrepresented among severe cases. Some, though not all, have found that people with blood group O appear to have a lower risk of the most dangerous forms of falciparum malaria, such as cerebral malaria and severe anemia, compared to people with A or AB. One proposed mechanism involves those sticky red blood cells. When falciparum parasites remodel the red cell surface, they can make infected cells stick to each other, as well as to blood vessel walls. In the lab, red cells from nonso individuals tend to form tighter clusters or rosettes around infected cells than O cells do. Larger, stronger rosettes are thought to clog vessels more easily, potentially contributing to severe disease. If that lab effect plays out in real infections, it could mean that in a heavily malarial environment, being type O gives a modest edge against the worst outcomes, not necessarily against infection itself, but against dying from it. In such a scenario, O might be slightly favored where falciparum has been intense for thousands of years. But the story is not simple, and it is not settled. Not all studies find the same patterns. Some show a protective effect of O in one region and not in another. Some find that A is associated with more severe disease in certain populations, while others find only weak or no associations. Many of the studies involve relatively small sample sizes, or they focus on hospital cases which may not represent the full spectrum of infection in the community. Even where a signal is fairly consistent that O is underrepresented among the most severe falciparum cases in a given region, the effect size is usually modest. Having type O does not make someone immune to malaria. Many O individuals still become infected, and some still develop severe complications. Likewise, being type A or AB does not doom someone to a bad outcome. These are risk modifiers, not fate. There is also the question of trade-offs. A blood type that helps a little with malaria might make you slightly more vulnerable to another pathogen or to a different kind of disease. Some research suggests that nonotypes may have advantages in other contexts, such as certain bacterial infections or bleeding risks. The full picture is a web of interactions, not a single clean adaptation. What most researchers agree on is more general. Malaria has been with humans and our ancestors for a very long time. Mosquitoes capable of carrying malaria are ancient. The parasites themselves show deep evolutionary histories. For tens of thousands of years in tropical and subtropical regions, generation after generation of humans have faced this threat, often in childhood. Selection acting over that kind of timescale doesn't have to be dramatic in each generation to leave a mark. Even small differences in survival or fertility repeated endlessly, can reshape gene SMAN frequencies. That is likely what happened with sickle cell and Duffy. It is plausible that more subtle shifts involving or other blood group systems also accumulated in certain regions. And this is where malaria intersects with the broader theme of polymorphism. If the parasite environment changes from place to place, different mosquito species, different plasmodium strains, different transmission intensities, and if various blood traits each confer small context-dependent advantages or disadvantages, then no single best blood type will win everywhere. Instead, different combinations will be favored in different settings. Add in genetic drift, founder effects, and other diseases that pull in opposite directions, and you get what we actually see, a patchwork of blood group frequencies shaped by overlapping histories. So, when we talk about malaria and blood types, it is tempting to reach for simple slogans. O is the malaria blood type, or this group is resistant. The reality is more measured. Some red blood cell variants show strong, clear protection and a heavy cost when inherited in double dose. Others, seem to exert gentler probabilistic effects that vary with geography and pathogen ecology. The lesson, perhaps, is methodological as much as biological. Correlations in the data can point us toward mechanisms like resetting or invasion pathways, but they're not proofs of destiny. The red blood cell is a battlefield, but it is also a crowded marketplace of interacting traits. Malaria is a powerful force, but it is only one of many combatants shaping the patterns we carry in our veins. And malaria is not the only disease that may have nudged those patterns in different directions. In other parts of the world, far from the mosquito-filled wetlands of the tropics, other pathogens have left their own signatures on the system. To see that, we have to leave the bloodstream and move into the gut to a different kind of epidemic pressure, diseases like cholera. If malaria turns red blood cells into a battlefield, cholera fights on a different front. It wages war on the lining of the gut. For most of human history, people didn't know the name cholera. They only knew what it did. Sudden explosive diarrhea, unstoppable vomiting, and a terrifying loss of fluid. A person who seemed healthy in the morning could be dead by nightfall. Their body collapsed, their skin cold and shriveled from dehydration. In some outbreaks, entire neighborhoods emptied in days. The disease has probably been with humans in some form for a very long time, but it was in the 19th century, with the growth of dense industrial cities and global trade routes, that cholera announced itself to the modern world. It erupted out of the Ganges Delta in South Asia, followed shipping lanes, and turned into a serial killer of continents. London, Paris, Hamburg, New York, Cairo, St. Petersburg. Waves of epidemics hit again and again. Cholera became a classic case for studying why some people get sicker than others. In the packed wards and makeshift hospitals of those epidemics, doctors could see that not everyone exposed to the disease died. Some were devastated, losing layers of fluid an hour. Others had milder illness, or none at all, even when they lived in the same crowded houses, drank the same contaminated water, and cared for the same patients. This unequal suffering raised an obvious question. Was there something intrinsic about the person? Some hidden trait that shaped how hard cholera hit? To connect that question to blood types, we have to shift our focus from the blood itself to the purposes that separate us from the outside world. The system is usually introduced as a property of red blood cells. AB, and O are described as different labels on those cells, built from tiny sugar structures. But the genes that control don't actually care whether a cell is in the blood or somewhere else. They are enzymes that modify certain basic sugar chains. And wherever those sugar chains appear, the A and B enzymes can act on them. That means antigens show up not just on red cells but on many other tissues, the cells that line blood vessels, the kidneys, and crucially for cholera, the epithelial surface of the intestines and other mucosal sites. In people who are secrets for another related gene system, these antigens are also present in saliva, mucus, and other bodily fluids. So when a pathogen arrives at a mucosal surface, say bacteria landing on the gut lining, the molecular landscape it encounters can differ subtly from person to person, depending on their type. The same base structure may be decorated with an A sugar, a B sugar, both, or left plain, as in type O. Cholera caused by the bacterium Vibrio is especially relevant here because it is not usually the sheer number of bacteria that kills. It is the toxin they produce. Cholera toxin latches onto receptors on the surface of intestinal cells, gets taken inside, and then flips on a molecular switch that drives massive secretion of water and electrolytes into the gut. The intestine, which usually reclaims water, suddenly becomes a fire hose. If tiny differences in the sugars and proteins on those intestinal cells influence how well the toxin can bind, they might influence how far that disastrous process runs. In the late 20th century, as blood typing became routine and cholera remained endemic in parts of South Asia and elsewhere, researchers started to look closely at whether type correlated with disease severity. They weren't asking who got infected, because in many outbreaks, almost everyone exposed seems to carry at least some bacteria. They were asking who landed in the most severe category: the people who needed intravenous fluids, who collapsed, whose lives hung on the speed of rehydration. Repeatedly, especially in places where cholera is long established, one pattern kept showing up. Individuals with blood group O often seemed more likely to experience severe dehydrating cholera than individuals with A or B. Not in every study, not identically in every outbreak, but often enough and with enough consistency in some regions that it drew attention. In Bangladesh, for example, several investigations over multiple epidemics have reported that among hospitalized cholera patients, type O is overrepresented, while among infected people with mild or no symptoms, no types appear more common. Similar trends have been described in other endemic areas. It is important to be cautious here. Cholera outbreaks are shaped by many things: the strain of Vibrio involved, the background health and nutrition of the population, patterns of immunity from earlier exposures, and simple chance. Not every study finds a strong effect, and some show only modest differences. As with malaria, these are risk gradients, not firm destinies. Type O people can have mild cases. No people can become desperately ill. Still, the signal is intriguing enough that scientists have tried to understand why it might exist. At the molecular level, choletoxin uses a specific handle to grab onto intestinal cells, a component of a ganglioid molecule called GM1. This is not itself an antigen, but the broader membrane environment. The neighborhood of lipids and sugars around GM1 can vary depending on which modifications are present. One plausible hypothesis is that in type O individuals, where the A and B sugars are absent, the arrangement of GM1 and surrounding molecules makes it easier for cholera toxin to bind tightly, cluster its receptors, and trigger the full catastrophic signaling cascade. In individuals with A or B antigens on their gut cells, the extra sugars might subtly interfere with that process, changing the spacing of receptors or the way they move in the membrane enough to dampen the toxin's effect. Experiments in cell cultures and animal models offer some support for this idea. Cells engineered to express different antigens can show differences in how well choletoxin binds or how strongly it activates signaling pathways. But these are models, simplified systems. Translating those findings into exact predictions about human disease severity in messy, real-world conditions is still an ongoing challenge. There is also a broader layer of interaction. ABO antigens on mucosal surfaces can influence the composition of the gut microbiome, the mix of commensal bacteria that live in the intestines. Some microbes stick more readily to certain sugar patterns and may help block pathogens simply by occupying space and consuming resources. If your type shapes which friendly bacteria thrive in your gut, it might indirectly affect how an invader like Vibrio behaves once it arrives. So we end up with a set of overlapping hypotheses. Choletoxin may bind more efficiently or set off stronger signals on the intestinal cells of certain types. The surrounding microbiome may differ in ways that help or hinder the pathogen. Other genes, like the secret status of the FUT2 gene, add more layers. None of these mechanisms has been nailed down to the degree of certainty we have for, say, the Duffy, negative protection against one kind of malaria. But together, they sketch a plausible map of how cholera and might be connected from an evolutionary perspective. Even a modest, consistent difference in mortality or fertility can matter if it is repeated often enough. Imagine a river in society, along the Bay of Bengal, or the Ganges Delta, 3,000 years ago. Every few decades, when the monsoon is bad, the water system is contaminated, and sanitation collapses. A cholera outbreak sweeps through. Children, the elderly, pregnant women, are especially vulnerable. Among them, perhaps type O individuals are slightly more likely to die or lose pregnancies when infected than their neighbors with type A or B. Now run that scenario, not just once, but hundreds of times across centuries. Each outbreak nudges the frequencies a tiny bit. Nonojopes gain a slight edge in survival and reproduction in that specific environment. In other regions where cholera is rare but malaria is intense, the balance might tilt the other way. Type O's potential advantages with severe falciparum disease could matter more than its possible disadvantages with gut infections. When geneticists look at global maps of frequencies, they see patchworks that do not line up with any single disease, climate, or culture. Instead, they likely reflect the cumulative effect of many pressures. Cholera in some places and times, malaria in others, typhoid here, smallpox there, plus drift, migration, and chance. Cholera's role in that story is still being pieced together. The evidence that it has seen the system, and may have pushed on it, is suggestive, but not definitive. The mechanisms that link tiny sugar groups on intestinal cells to the violence of an epidemic are still being refined. But the broader lesson is quietly unsettling. The difference between type O and type A, food one sugar decoration and another, feels vanishingly small at the scale of a molecule. Yet in the middle of a 19th century slum, or a modern refugee camp with contaminated water, that tiny variation might change how long you have to reach clean fluids, how likely you are to survive the night. And cholera is only one example. Other pathogens, some infamous and some now largely forgotten, may also have exploited our blood group chemistry in their own ways, leaving their fingerprints on the distribution of A, B, and O. To follow those traces, we have to step out of the gut and into the history of some of the world's most feared infections, smallpox, plague, and the many other epidemics that have tested the resilience of our blood. When historians list the great killers of the last few thousand years, cholera is only one line in a grim catalogue. Smallpox, plague, typhoid, influenza, measles, tuberculosis. Again and again, new or newly empowered pathogens have swept through human populations, reshaping societies and leaving demographic scars that can still be seen in records, ruins, and sometimes in our genes. From the perspective of a geneticist, each of these episodes is not just a tragedy. It is also coldly a selection event. Whenever a disease is common enough, deadly enough, and persistent enough, it has the potential to favor some genetic variants over others, not in a simple conscious way, but through the brutal arithmetic of survival and reproduction. If people with a particular variant are just a little less likely to die young, or a little more likely to have surviving children, that variant can slowly rise in frequency over generations. This is why researchers keep turning back to blood groups when they think about historical epidemics. The system and related blood group genes are easy to type, vary dramatically across populations, and sit right at the interface between our cells and the microbes that try to exploit them. If any part of our genome has been pushed around by repeated waves of infectious disease, is a prime candidate. One of the most intensively studied suspects in this story is smallpox. For at least 1,000 years and probably longer, smallpox was one of humanity's most reliable executioners. In some societies, it killed 1 in 3 children who caught it. Those who survived often carried deep, pitted scars and sometimes blindness for the rest of their lives. In the era before vaccination, few people reached adulthood without encountering it, directly or indirectly. Unfortunately for scientists trying to reconstruct its evolutionary impact, the virus that caused classical smallpox, variola major, is now gone from the wild. It was eradicated in the late 20th century by vaccination campaigns, and surviving samples are locked away in high-security labs. That means we cannot run large modern case control studies the way we might for cholera, malaria, or COVID-19. Instead, researchers have had to be creative. They use a mixture of old medical records, data from related pox viruses, historical descriptions of outbreaks, and statistical patterns in modern blood group distributions. As early as the mid-10th century, some physicians noticed hints that blood type might influence how people responded to smallpox vaccination or to natural infection. A few early studies suggested that individuals with type A might develop more severe skin reactions to certain pox viruses, while those with type O or B had milder courses. Others reported different or weaker trends, or none at all. Why the inconsistency? Partly because the data were messy. Many of these studies were small, conducted under wartime or outbreak conditions, and used different criteria to define mild versus severe. They often lacked detailed information on age, nutrition, prior immunity, and co-infections, factors that can heavily shape how any infection plays out. More recently, as genetic tools improved, some population-level analyses found signals that parts of the genome around the region show signs of path selection that could in principle be related to pox virus exposure. Variola and its relatives are adept at interacting with host immune molecules on cell surfaces, and the idea that they might have seen antigens is not far-fetched. But crucially, none of this adds up yet to a clear, simple statement like type X was strongly protected against smallpox. Instead, we have suggestive patterns, plausible mechanisms, and a lot of uncertainty. Plague, the disease behind the Black Death in 14th century Europe, and many other outbreaks, before and after, is similar in that way. The bacterium Yersinia pestis has a complicated life cycle involving fleas, rodents, and humans. When it spills over into people under the right conditions, it can cause extraordinarily high mortality in a short time. In some medieval European towns, half or more of the population may have died within months. This kind of shock is exactly the sort of event that could, in theory, reshape genetic variation. And indeed, modern studies have found signs of strong selection around certain immune-mediated genes in Europeans that may date to the time of the Black Death. When it comes to researchers have looked for links between blood type and plague susceptibility using a mix of approaches. Some have analyzed records from later outbreaks when partial blood typing was available to see whether certain groups were overrepresented among the dead. Others have used modern data from regions where Yersinia pestis still circulates, comparing blood group frequencies in people with a history of plague exposure versus those without. A few laboratory studies have explored how Yersinia interacts with cells bearing different surface sugars. The results are again mixed. A few analyses have hinted that certain nonotypes might have fared differently during some outbreaks. Others see no significant association. The sample sizes are often small, the historical diagnosis uncertain, and the social context complicated. Who was exposed, who could flee, who was buried where, all depended on status, occupation, and geography. Not just biology, these problems are not unique to smallpox or plague. For many historical diseases, typhoid fever, meningococcal infections, different strains of influenza, even some forms of viral hepatitis, there are scattered reports of associations. Type O might be more prone to severe disease here, type A there, type B somewhere else. Sometimes these match lab findings about how a pathogen binds to cells or evades immune molecules. Sometimes they don't. So why is the evidence so variable? Part of the answer lies in the pathogens themselves. Many infectious agents, especially RNA, viruses, and some bacteria, evolve quickly. The strain of influenza that circled the globe in 1918 is not the same as a seasonal flu strain today. Cholera in 19th century London was not genetically identical to modern strains in Bangladesh. If a particular pathogen once exploited a specific blood group antigen efficiently, a later strain might not. Another part lies in the historical record. Before the mid-10th century, Systematic blood typing of entire populations was rare. Many proposed associations are reconstructed retrospectively, using incomplete data or inferred indirectly from death registers, military records, or localized surveys. People often classified illnesses with similar symptoms. Plague might in some cases have been typhus. Fever could have meant a dozen different things. Then there are confounders. Blood group frequencies are not uniform across ethnicities or regions, and neither are wealth, diet, housing quality, or access to care. If type B is more common in a community that also happens to live in crowded tenements next to contaminated water, it may look more susceptible in crude statistics, even if the real driver is environment, not chemistry. Modern epidemiology tries to untangle these threads by using careful study designs. Researchers compare cases and controls matched for age, sex, and background. They adjust statistically for known confounders. They look at large sample sizes to avoid being misled by random noise. But even now, not every study can account for every factor, and results can still disagree. To move beyond correlations, scientists combine these population studies with laboratory work and genetics. In the lab, they microcells engineered to display different antigens, then expose them to a virus or bacterium to see which ones it sticks to, enters, or kills more readily. They test whether antibodies against certain blood group sugars can block infection. They study how complement proteins and clotting factors, which are also influenced by behave during sepsis or viral inflammation. On the genetic side, they scan the genomes of large numbers of people looking for footprints of past selection, unusual patterns of variation around the locus that suggest a strong, relatively recent push. Then they try to line up the estimated timing of that selection with known disease histories. Did a big shift in frequencies appear around the same era as a major epidemic in that region? Is there a plausible biological connection between the pathogen, lifestyle, and the antigens it might encounter? Taken together, these approaches often produce a mosaic of evidence rather than a single sharp picture. For some pathogens like Plasmodium falciparum malaria, the pieces line up well. Epidemiology, mechanism, and selection signals all point in the same direction. For others, like cholera, the pattern is suggestive but not yet conclusive. And for many historical killers, Smallpox and plague among them, the trail is thinner, interrupted by gaps in data, changes in the pathogens themselves, and the sheer complexity of human societies. This is why most specialists are cautious. They suspect that smallpox, plague, and a host of lesser-known infections did interact with other blood group systems in important ways. They see signs that these diseases may have nudged the frequencies of A, B, and O over centuries, but they're reluctant to point to any one pathogen and declare it the single architect of our modern blood type map. Instead, a different image has emerged. Imagine the last 10,000 years not as a single battlefield with one dominant enemy, but as a long series of overlapping campaigns. In one region and century, smallpox burns hottest, and any tiny advantage against it matters. In another, plague flares repeatedly from rodent reservoirs. Elsewhere, cholera, typhoid, and dysentery thrive in crowded river towns. While in yet another zone, malaria remains the primary killer of children. Each of these diseases may read the sugars on our cells in slightly different ways. Some may find type O easier to exploit. Others may struggle more with type B or AB. A few may be largely indifferent to, but care deeply about other blood group systems or immune genes. Meanwhile, migrations, bottlenecks, and simple chance shuffle the deck of algils as people move, mix, and sometimes vanish. The result is the patchwork we see today. High B frequencies in parts of Asia, higher frequencies in the Americas, diverse mixes across Africa and Europe. Not the fingerprint of one pathogen, but of many. A mosaic of selective pressures layered on top of demographic history. When we look at that mosaic, we can sometimes pick out individual tiles. Malaria here, cholera there. For smallpox, plague, and other historic horrors, the tiles are more faded, the outlines blurrier. Some connections that seem convincing now may weaken as new data arrive. Others that look vague today may sharpen with better ancient DNA, more complete outbreak records, or new laboratory models. What remains constant is the underlying principle. For thousands of years, our blood group antigens have been part of a shifting conversation between human cells and the microbes that challenge them. Smallpox, plague, and the many other diseases our ancestors endured were all participants in that conversation, even if we cannot yet reconstruct every word. And blood groups themselves are not just passive targets. Those same tiny sugar differences that might tilt the odds in an infection can also influence clotting, inflammation, and how our own tissues interact with each other. As we move beyond specific pathogens, another layer of selection begins to appear, one that comes not from dramatic epidemics, but from the quieter everyday risks of bleeding, pregnancy, and chronic disease. When scientists talk about disease pressures, it's easy to picture only the obvious villains: microbes, parasites, waves of infection sweeping through a village. But the body itself can be an unforgiving environment. Every day, without any help from bacteria or viruses, your blood has to perform a series of delicate balancing acts. It needs to clot quickly enough to stop you from bleeding to death if you cut yourself, but not so eagerly that it blocks your arteries. It has to mount inflammation to clear damage, but not so much that it starts destroying healthy tissue. It has to allow cells to stick and unstick from vessel walls in a controlled way. ABO and Rh are involved in those balances too. One of the most consistent non-infectious associations has to do with clotting factors, especially a protein called von Willebrand factor and a related clotting factor known as factor VIII. People with non-blood types, particularly A and B, tend to have higher average levels of these proteins circulating in their blood than people with type O. On the face of it, that might sound like an obvious benefit. More clotting factors should mean faster, more reliable clotting. In a world without hospitals, where a deep cut or childbirth hemorrhage could be fatal, that extra margin might have been genuinely protective. But clotting is a double-edged sword. The same higher levels of von Willebrand factor and factor VIII are linked in modern populations to a slightly increased risk of forming dangerous blood clots inside blood vessels. Conditions like deep vein thrombosis and pulmonary embolism, no individuals on average show higher rates of these events than type O, even after you account for some other risk factors. The key word here is slightly. These are statistical tendencies that show up when you look at thousands or millions of people. They do not doom any individual with type A or AB to a heart attack, and they do not magically protect every type O person from cardiovascular disease. Lifestyle, diet, smoking, blood pressure, exercise, and simple aging all loom much larger in determining whether a particular person will develop a clot or blocked artery. Still, the patterns are real enough that cardiologists and hematologists pay attention to them. Large genetic studies have repeatedly found that variants in and around the gene are associated with the risk of coronary artery disease and stroke. These effects are modest compared to, say, heavy smoking, but they are part of the background landscape that shapes risk. In an evolutionary context, this kind of trade-off begins to look familiar. Imagine an environment where people frequently suffer traumatic injuries from hunting, falling, or childbirth, and where medical help is essentially non-existent. In that context, a blood type that tilts you slightly toward faster clot formation might be favored. Despite the eventual cost of more clots in middle or late adulthood, if many people never lived long enough for age-related cardiovascular disease to matter, the benefit in youth would outweigh the harm in old age. Now imagine a different environment, perhaps one where infectious diseases that trigger massive inflammation are common, or where diets and lifestyles combine with that clotting tendency to produce early heart attacks. In that context, the same blood type might look less advantageous. This is the essence of a trade-off. The trait is not good or bad in some absolute sense. Its value depends on what the world throws at you, and at what age. ABO and Rh may also be involved in less obvious but equally important negotiations within the body. especially during pregnancy. We already explored Rh incompatibility, where an Rh-negative mother carries an Rh-positive fetus, and in some pregnancies, mounts an immune response against fetal red cells. That is a dramatic, well-documented example of maternal and fetal blood traits coming into conflict. Beyond that, there is a subtler web of interactions that researchers are still untangling. The placenta, the temporary organ that connects mother and fetus, is a zone of intense biological diplomacy. Maternal blood bathes fetal tissues. Nutrients, hormones, and signaling molecules move back and forth. Tiny differences in cell surface molecules, including some related to blood groups, can potentially influence how smoothly that exchange proceeds. Some studies have suggested that certain combinations of maternal and fetal types might affect the risk of complications like preeclampsia, fetal growth restriction, or miscarriage. Others have explored whether or Rh status shifts the odds of particular pregnancy outcomes in ways that are too small to notice at the level of a single family, but measurable in large datasets. So far, the picture is hazy. Results differ between populations and study designs. Many apparent effects vanish when you control for age, prior pregnancies, nutrition, or access to care. A few associations persist, but they are generally modest. Again, nothing in this research justifies viewing a particular blood type as a sentence of reproductive difficulty or as a guarantee of smooth pregnancies. What it does suggest is that maternal-fetal interactions may have contributed in quiet, incremental ways to the evolutionary story of blood groups. If certain mother-child combinations slightly improved the odds of a successful birth in one ecological or cultural context, those combinations might leave a faint imprint on the gene pool over thousands of years. As we add up infections, clotting, cardiovascular disease, pregnancy, and probably other processes we've not yet recognized, another concept from genetics becomes crucial: pleiotropy. Pleiotropy is the idea that one gene can influence many seemingly unrelated traits. ABO is a classic example. The same gene that determines whether your red cells carry A, B, or O. Antigens also affects which sugar chains decorate cells lining your blood vessels, your gut, and other tissues. Those sugar patterns can influence how pathogens attach, how clotting factors behave, how immune molecules circulate, and how cells signal to one another. From the point of view of natural selection, this means that a change at the locus is never just about one thing. A variant that slightly reduces the risk of severe cholera might simultaneously raise the risk of a particular kind of thrombosis. A change that helps defend against one virus might alter how the placenta handles maternal antibodies. A tweak that affects clotting could, in turn, feed back into how the body responds to sepsis. Rh is less pleiotropic in terms of obvious systemic effects, but even there the story is likely larger than the simple presence or absence of the classic deant antigen. The Rh complex is involved in the stability of red cell membranes. Subtle differences could influence how long red cells survive, how they respond to oxidative stress, or how they behave in microcirculation. All of this complicates any attempt to tell a single clean evolutionary story. There is no reason to think that one great plague, one dietary shift, or one reproductive challenge designed our current mix of blood types. Instead, what we see looks more like the accumulated result of many small overlapping forces, some infectious, some not, acting on genes that touch multiple aspects of physiology at once. Over tens of thousands of years, as human groups moved into new climates, adopted new foods, faced new diseases, and developed new social and reproductive patterns, the balance of advantages and disadvantages for each blood type kept shifting. In one time and place, type O might offer a bit more protection against malaria at the cost of more severe cholera. In another, type A's clotting profile might be favored in a harsh, injury-prone environment, even if it nudged cardiovascular risk later on. Elsewhere, B or AB might interact with local pathogens, diets, and reproductive norms in their own distinct ways. Meanwhile, chance events, bottlenecks, founder effects, migrations were constantly reshuffling the deck. A small band of people carrying a particular variant might happen to colonize a new region and grow not because their blood type was superior in any global sense, but because they arrived first and survived well enough. When we look at modern associations between blood types and non-infectious conditions, heart disease, stroke, bleeding disorders, pregnancy complications, it is tempting to read them as direct reflections of past selection. They may be part of that story, but they are also shaped by the contemporary world, by processed foods, sedentary lifestyles, medical technologies that allow many more of us to reach ages at which chronic disease becomes relevant. In that sense, the same blood group of R now playing out in a very different theater than the one in which they were originally tested. Keeping this in mind helps us avoid two extremes. On the one hand, we should not dismiss these associations as meaningless just because they are modest. Small differences in risk can matter at the level of populations and over evolutionary time scales. On the other hand, we should not inflate them into destiny. Your blood type is one variable among many, and for most modern individuals, it is not the dominant driver of health outcomes. The persistence of multiple blood types in our species then probably reflects no single, dramatic explanation. Instead, it points to a long history of compromise, genes with multiple effects being tugged in different directions by bleeding risks and clotting risks, by infections and inflammation, by maternal-fetal negotiations and the hazards of aging. To see how deep that history runs and how these tiny differences have moved with us across continents and millennia, we have to look not only at living people, but at those who left their DNA behind long ago in bones, teeth, and frozen soils. Ancient DNA is our way of eavesdropping on those long-vanished gene pools. When scientists pull fragments of DNA out of a tooth or a bit of petrous bone and run them through modern sequencing machines, they're not just recovering a single genome. They're catching a faint time-stamped snapshot of how genes were arranged in a particular place, in a particular person, at a particular moment in the past. For blood groups, that is both incredibly powerful and inherently limited. On the powerful side, the ABO and Rh systems are determined by very specific genetic changes. At the locus, we can recognize the classic AB and O variants from their characteristic mutations in the gene that codes for a sugar-adding enzyme. At the RHUS, we can identify the presence or absence of the RD gene and many of the subvariants that tweak Rh expression. If a sample is well preserved and if enough of those key stretches survive the ravages of time, we can say with reasonable confidence this individual who lived 5,000 or 10,000 or 40,000 years would have been type A or O or B and either Rh positive or Rh negative. But that is where the limitations begin. A single skeleton is not a population. A handful of sequenced burials from one cave or one cemetery do not fully represent the living community from which they came, let alone all the people who lived across a continent at the same time. Ancient DNA datasets are also deeply biased by preservation. Cold, dry, stable environments are kind to DNA. Warm, wet, acidic ones are not. So, our picture of past blood types is skewed toward places where bone and tooth survive. European caves and burial grounds, Siberian permafrost, high-altitude deserts, a few exceptionally dry sites in the Near East and the Americas, huge depots of the world's past populations, tropical Africa, much of South and Southeast Asia, land rainforest regions are either underrepresented or almost invisible in the ancient DNA record simply because their remains do not preserve as, or have not yet been sampled. Then there is the human layer of bias. Archaeologists excavate certain sites more intensively than others. Museums decide which bones to curate. Geneticists choose which samples to sequence. Often we have more data from elites buried in monumental graves than from ordinary people whose remains were never preserved. All of that means that when we talk about ancient blood type frequencies, we are really talking about glimpses, patchy, uneven glimpses through the fog of time. Still, those glimpses are starting to outline a story. If you follow and Rh variants through the ancient DNA record, you see that the modern patchwork of blood types did not arise recently. It has deep roots that stretch back through the ice ages and the great movements of people that followed. Take the last glacial maximum, roughly 20,000 years ago. Much of northern Europe and North America lay under massive ice sheets. Conditions were harsh, and human populations in many regions shrank and retreated into more habitable pockets. Ice Age refugia in southern Europe, the Caucasus, parts of the Near East. Within those refuges, small groups of hunter-gatherers survived, reproduced, and slowly accumulated their own combinations of genetic variants, including and Rh alleles. Some refugal groups might by chance have had more O alleles, others more A or B. There is no reason to think the distribution was uniform, even over relatively short distances. It almost certainly was not. Then, as the ice sheets melted and the climate warmed, people expanded out of those refugia, reoccupying northern Europe and other formerly glaciated lands. When they moved, they carried their blood group profiles with them. A valley repopulated by descendants of one refugal group would end up with a slightly different mix of blood types than a region colonized by another. Later, another wave pushed through. The Neolithic expansion of farmers. Starting in the Fertile Crescent and parts of Anatolia around 10,000 years ago, populations that had adopted agriculture began to grow rapidly and spread into Europe, North Africa, and parts of Asia. Ancient DNA has shown that these Neolithic farmers did not simply teach local hunter-gatherers how to farm. They often replaced or heavily admixed with them. As those farming groups fanned out, they again carried particular clusters of and Rh alleles. In some regions, they mixed with local hunter-gatherers who had different blood type distributions, creating new blends. In others, their descendants largely replaced the earlier inhabitants, leaving a different genetic signature. What we see today in many parts of Europe, the relatively high frequency of type O, in some areas of A, in others, the comparatively low B, frequency in the far west, compared to Central and South Asia, is the product of those repeated cycles of contraction, refuge, expansion, and admixture layered on top of the selective forces we talked about earlier. Similar stories played out elsewhere, although the details differ. In parts of East Asia, the expansion of early rice and millet farmers reshaped the gene pools that have been established by Paleolithic hunter-gatherers. In the Americas, the initial peopling from Siberian ancestors carried a particular set of and Rh variants across the Bering region, where small founding populations and subsequent bottlenecks would have magnified chance fluctuations in blood type frequencies. Later demographic events, the rise and fall of complex societies, migrations, epidemics introduced after European contact, added further reshaping. Each of these episodes acts like a hand, stirring different colored sands together. Sometimes one color becomes dominant in a region, not because it is inherently better, but because of who arrived, who stayed, and who had children. Importantly, ancient DNA also undercuts the simple idea that ancient equals homogeneous. Even within a single time slice, say Bronze Age Europe or pre-Columbian Mesoamerica, the genetic evidence points to regional and sometimes very local diversity. Neighboring valleys, coastal versus inland communities, herring groups versus settled, farmers could have subtly different blood type profiles, just as they had different languages, customs, and diets. Blood groups were part of that fine-grained mosaic. They were not a single uniform signature stamped across a continent, but one more layer of variation, drifting and shifting as people moved, mixed, split, and reunited. The art system shows these patterns too, though in its own way. The classic distinction between Rh positive and Rh negative largely reflects whether the Rhd gene is present or deleted, and that deletion seems to have reached particularly high frequencies in some West Eurasian populations. Ancient DNA suggests that the spread of negative alleles through Europe involved the same kinds of processes, bottlenecks, expansions, and mixture rather than a sudden mysterious appearance. All along, sampling bias hovers in the background. When we map ancient blood types, we are mapping the people whose bones happen to fall into the right soil, whose graves happen to be excavated, whose remains happen to be selected for analysis, and whose DNA happen to survive. Entire populations can rise, thrive, and vanish while leaving almost no recoverable genetic trace. So the shifting map we can draw today is best understood as a sketch, not a finished atlas. Lines are missing, regions are blank. Some features are drawn in thick ink, others only as dotted hints. Yet even in this incomplete form, the picture changes how we think about blood types. Instead of static labels printed on a hospital card, and Rh become quiet markers of deep history. Each allele Ucarus traveled through a chain of ancestors who weathered Ice Age winters and Holocene droughts, who may have huddled in glacial refugia or walked behind the first, domesticated herds who took part, whether they knew it or not, in the great demographic waves that swept across continents. In your veins, the consequences of those movements are still present, not in any mystical way, but in the literal arrangement of the sugars on your red blood cells. To see just how far back that ancestry reaches and how other human species contributed to this story, we have to widen the lens beyond Homo sapiens alone and follow and Rh back into the world of Neanderthals, Denisovans, and the other cousins with whom we once shared the planet. When geneticists talk about Neanderthals and Denisovans living on in us, they are not speaking metaphorically. For a small but meaningful fraction of our genome, we can point to specific stretches of DNA and say, this piece didn't just arise in Homo sapiens, it was borrowed long ago from another kind of human. Many of those stretches are involved in immunity. Across the genome, regions that help recognize pathogens, present foreign molecules to immune cells, or regulate inflammatory responses, show especially strong evidence of archaic introgression. In other words, Neanderthal and Denisovan versions of certain immune genes seem to have been picked up by our ancestors and kept because they were useful. Once you see that pattern, it is natural to ask, what about blood groups? After all, the molecules that define and many other blood group systems sit right on the surface of red blood cells and other tissues. They are part of how our bodies interface with pathogens, how our immune systems recognize self and non-self. If Neanderthals and had useful variants at other immune loci, maybe some of our blood group diversity is their legacy too. This is exactly the kind of question researchers have been exploring over the past decade as more high-quality archaic genomes have become available. The first step is simply to ask, what blood types did these other humans likely have? For Neanderthals, we now have several reasonably complete genomes from individuals who lived in Europe and Western Asia, plus a few lower coverage sequences. When scientists examine the gene in those genomes, they find recognizable forms, variants that correspond to A-like, B-like, and O-like alleles. In some Neanderthals, the sequence looks very similar to modern nonnuls, suggesting they would have expressed A or B antigens. In others, the gene carries frameshift or nonsense mutations, characteristic of OIP alleles, that do not add the defining sugars. Denisovan genomes tell a similar story. Their gene is not identical to ours, but it uses the same basic machinery, an enzyme that adds sugars to a precursor molecule with different versions producing different terminal sugars, and loss-of-function versions leaving the precursor unmodified. So at a very broad level, it appears that Neanderthals and Denisovans had ABO polymorphism too. They were not fixed for a single blood type. They carried multiple alleles at the locus, just as modern humans do. From there, the question becomes more subtle. When we see a particular variant in both Neanderthals and modern humans, how did that sharing arise? There are three main possibilities. One is shared ancestry. The polymorphism could be old enough that some of its variants were already present in the common ancestor of Neanderthals, Denisovans, and modern humans hundreds of thousands of years ago, and have simply been carried along in multiple lineages ever since. Another is introgression. A particular Neanderthal, or Denisovan allele, might have entered the Homo sapiens gene pool during interbreeding events in Eurasia, and then persisted at low or moderate frequency. The third is convergent evolution. Similar selective pressures could, in principle, produce similar functional variants, independently, in different lineages, especially at a locus where the same few positions can be tweaked in similar ways. Distinguishing among these scenarios is not always easy. In some cases, the broader genomic context points strongly to introgression. If a chunk of DNA around a blood-related gene looks distinctly Neanderthal-like, matching Neanderthal sequences much more closely than African modern human sequences, and if that chunk is found only in non-African populations today, the simplest explanation is that it arrived via interbreeding after modern humans left Africa. Several immune loci fit this pattern clearly. For blood group systems, the evidence so far is more patchy. For example, some studies have identified Neanderthal-derived haplotypes near genes involved in red blood cell antigens and coagulation. These segments appear to influence traits such as clotting risk or susceptibility to certain infections, and they carry telltale signatures of archaic origin. That makes them good candidates for adaptive introgression. Inherited from Neanderthals, because they conferred a survival advantage in Eurasian environments. At the locus itself, the picture is more mixed. Certain rare or geographically restricted HLET types in modern populations resemble the form seen in Neanderthals more than the predominant modern variants. Some researchers have suggested that these could represent Neanderthal-derived alleles that slipped into our gene pool and survived, perhaps under balancing selection that favors diversity at this site. But because polymorphism is so ancient, and because the same functional changes can arise more than once, it is difficult to be definitive. A Neanderthal-like sequence in a modern human might be archaic in origin, or it might be an old shared variant, or it might be a similar solution reached twice under similar evolutionary pressures. For Denisovans, data even thinner. We have fewer genomes from fewer places, and their modern genetic legacy is concentrated in specific regions, most notably parts of East and Southeast Asia and Oceania. There is intriguing evidence that some blood-related traits in these populations, such as adaptations to low oxygen at high altitudes, are linked to Denisovan introgression around genes that affect red blood cell function. But clear examples involving the classic or Rh systems remain tentative. Rh in particular is technically harder to reconstruct from fragmentary ancient DNA, because the RHD region is structurally complex and prone to deletions and duplications. So while we can detect some Neanderthal and Denisovan contributions to broader red blood cell biology, the fine-grained story of Rh variants in archaic humans is still emerging. Amid these uncertainties, one conclusion has become steadily more secure. ABO is old, very old. When scientists compare the gene across primates, chimpanzees, gorillas, orangutans, and several monkey species, they find clear A-like and B-like variants, and in some cases O-like loss-of-function alzas as well. The molecular signatures that define A and B in humans are echoed in these other primates along very similar lines. The simplest explanation is that a form of polymorphism has been maintained in primate lineages for tens of millions of years, not always with exactly the same alzals and not always under exactly the same selective pressures, but with the same basic pattern. Multiple versions of the sugar-adding enzyme coexisting in populations for far longer than you would expect if one were simply best and the others were destined to vanish. This kind of long-term maintenance is classic balancing selection. It suggests that different types have had different advantages and disadvantages in different ecological and pathogen environments, and that no single version has ever been universally superior for long enough to erase the others. Seen from that perspective, Neanderthals and Denisovans were not exotic outliers in our blood group story. They were participants in a much older pattern. They inherited a polymorphic system from ancestors who were already playing this evolutionary balancing act. They may have added their own twists, unique variants, local adaptations, perhaps even contributions to our own gene pool at immune-irradiated sites before disappearing as distinct populations. Modern humans, for our part, carry a mosaic genome, mostly shaped within our own lineage, but with tiles contributed by these archaic cousins at some points and by much older primate evolution at others. When you look at your blood type against that backdrop, it becomes less a label pinned to a single species and more a faint echo of deep time. The A, B, or O you carry might trace its functional logic back to early apes. Some of the immune nuances that travel with it may have been sharpened in Neanderthal caves or Denis mountain camps. Others arose entirely within Homo sapiens, in farmers' villages or hunter-gatherers' camps far more recently. Untangling those layers is an ongoing project. Each new archaic genome, each better-resolved ancient sequence, adds a tiny bit of clarity. For now, we can say with some confidence that parts of the immune landscape around our blood groups bear the imprint of contact with other human species, and that the system itself is far older than Homo sapiens. But which specific variants in your veins are Neanderthal gifts, which are Denisovan legacies, which are shared inheritances from ancient primates, and which are purely our own inventions? That remains an open question. In other words, when we talk about our blood types, we may be telling a story that began long before there was anything on Earth that we would recognize as us. By the time scientists understood that these ancient, tangled histories lay behind something as ordinary as a blood type, the way we use that knowledge had already begun to change the modern world. At first, blood groups were a strictly practical discovery. In the early 20th century, when physicians started to grasp the system, transfusion medicine was still a dangerous improvisational practice. Surgeons knew blood loss killed patients. They knew that giving blood sometimes saved them and sometimes killed them faster. They did not yet know why. Once compatibility testing became possible, that changed quickly. Hospitals began to type patients before surgery. Small in-house blood banks formed, storing carefully labeled bottles from local volunteers. Instead of gambling on any donor who happened to be available, doctors could match O to anyone and avoid catastrophic reactions. It was a quiet revolution. No fireworks, no moment where the world stopped and gasped. Just chart after chart, where died on the table, was replaced by recovered without incident. Then came war. During the First World War, and even more during the Second, the logistics of blood supply became a military problem. You suddenly needed not just a few pints for a hospital, but rivers of compatible blood for thousands of wounded soldiers in field hospitals and surgical tents far from home. Blood typing moved out of research laboratories and into recruitment offices, aid stations, and mobile collection units. Technicians were trained by the tens of thousands. Simple, robust tests were developed that could be done quickly under rough conditions. Standardized systems appeared. Glass bottles replaced ad hoc tubing. Citrate solutions allowed storage. Refrigerated trucks carried precious units across battlefields. Along the way, people began to carry their blood type on paper tags, on metal discs, on cards tucked into uniforms. For many of those soldiers, that little code, A positive, O negative, B positive, was the first time their blood group had become part of how they imagined themselves. It was no longer just a laboratory result. It was stamped onto their dog tags, part of their official identity in a world where a few minutes could make the difference between life and death. After the wars, that infrastructure did not vanish. It came home. Civilian blood banks expanded. National and regional systems for collecting, testing, and distributing blood were built on top of the wartime experience. Hospitals integrated blood typing into routine admissions. Expectant mothers were typed as part of prenatal care, especially after the Rh factor and hemolytic disease of the newborn were understood. Then something new happened. Charities and health systems realized that blood wasn't just a medical commodity. It depended on people, on volunteers who would come in, roll up a sleeve and give. And blood drives became community events at schools, workplaces, churches, factories. People walked away not only with a bandage and a cookie, but with a little card in their wallet that said, clearly, you are O positive or you're AB negative. Over time, that simple, practical piece of data, the kind of thing a technician might once have scribbled in a lab notebook, started to feel like a label, a category, a small but crisp answer to the question, what are you? That feeling only grew as blood typing spread beyond hospitals. In many countries, students learned their type in school physicals. Some government ads added it. Military conscription made it nearly universal in certain populations. Once a trait is widespread, easily measured, and reduced to a handful of letters and symbols, it becomes almost irresistible as a hook for identity. Humans have always done this. We gravitate towards simple categories: zodiac signs, birth order, handedness, 4 temperaments, introvert versus extrovert. The categories don't need to be especially accurate. They just need to be memorable, easy to talk about, and flexible enough that people can see themselves in them. Blood type, with its short codes and clear-cut alternatives, fit the pattern perfectly. In some cultures, especially in parts of East Asia, this tendency crystallized into elaborate narratives. In Japan, for example, the idea that blood type reflects personality emerged in the early 20th century, was amplified by popular books in the 1970s and 1980s, and has lingered ever since. In these narratives, Type A people are often described as meticulous and earnest, Type B as creative and free-spirited, Type O as confident leaders, Type A as complex or eccentric. Similar stories circulate in South Korea and elsewhere, showing up in magazines, TV shows, even matchmaking profiles. Scientifically, there's no robust evidence that type predicts personality in this way. Large studies that compare blood type with psychological traits consistently find tiny, inconsistent effects at best, exactly what you would expect from random variation and confirmation bias. But socially, the narratives are real. They function like any other folk taxonomy of character. People joke about their type, blame it for quirks, use it as a light-hearted way to talk about compatibility. Over time, those stories can even become self-fulfilling as individuals lean into traits they believe fit their type. Behind this, it's a deeper pattern. Our brains like clean lines. The world and our biology are not clean. A system like is actually messy. Dozens of rare subtypes, partial antigens, interactions with many other blood group LOI. The Rh system alone includes multiple antigens beyond the famous D. Your red blood cells carry a mosaic of markers from at least 30 recognized blood group systems, but that complexity is hard to hold in mind. I'm B positive is easy. We use such labels to reduce a tangled web of evolutionary history, molecular biology, and statistical risk into something we can tell stories about. Along the way, we risk confusing the sliver of reality the label captures with the whole person. It is worth stepping back and asking, what can your blood type actually tell you? On the practical side, quite a lot. ABO and Rh status are critical for transfusion safety. They determine which donated units you can receive without provoking a dangerous immune reaction. They matter in pregnancy when an Rh-negative mother carries an Rh-positive fetus, because maternal antibodies can cross the placenta and harm fetal red blood cells, a risk that modern medicine manages with testing, monitoring, and Rh immunoglobulin. Blood type can also nudge the probabilities of certain medical events. As we saw earlier, nonotypes might have slightly higher risks of some clotting disorders. Type O may be marginally more vulnerable to certain infections and less to others. These are population-level tendencies, not destinies, but they are real enough to interest epidemiologists. Beyond that, the predictive power drops off quickly. Your blood type does not reliably determine your intelligence, your temperament, your romantic compatibility, your career prospects, or your moral character. It is one tiny axis of variation in a human genome that contains billions of base pairs shaped by environments, cultures, and choices that cannot touch. Yet, the myths persist, and new ones continue to arise. They do so for familiar reasons. Stories about types give us a way to navigate social life, to sort an overwhelming flood of information into manageable boxes. They offer a sense of belonging. You and your fellow O-types, or your fellow ABS, are part of the same small club. They provide ready-made explanations for behavior, flattering or otherwise. And because blood type is anchored in something undeniably real, a test result, a medical chart, it can feel more grounded than, say, star signs. The fact that the biological meaning and the cultural meaning do not line up neatly does not weaken the stories. It may even make them more alluring. In the next part of this journey, we'll follow those stories directly. We'll look at where blood type folklore came from, how it spread, and why, in a world full of complex genetics and careful statistics, simple tales about and AB still have such a powerful hold on the human imagination. When people first began to spin personality tales around blood types, they weren't doing molecular biology. They were doing what humans have always done when faced with something invisible yet powerful. They told stories. In Japan, in the early 20th century, as Western science and medicine were being rapidly imported and adapted, blood type was a new, modern, slightly mysterious idea. Here was a trait that could be measured in a laboratory, written down in crisp Latin letters, and tied directly to life and death on the operating table. At the same time, older traditions of divination and character classification were still very much alive. Systems that sorted people into types based on birth year, elements, or animal signs already had deep cultural roots. It didn't take much imagination to connect the two. In 1927, a Japanese educator named T.C. Furukawa published a short article suggesting that differences between national and ethnic groups might be linked to their distribution. His claims were speculative and deeply flawed, reflecting the prejudices of his time, but the seed was planted. Blood type as a window into character. The idea went quiet for a while, then returned decades later in a new, more domesticated form. In the 1970s, popular books by Masahiko Nomi took the concept of blood type personality and recast it as a kind of friendly everyday psychology. Nomi wasn't a physician or a geneticist. He was a writer who loved patterns. He collected anecdotes, interviewed people, looked at workplace dynamics, and convinced himself that the letters on a blood card lined up with the temperaments he thought he saw around him. For many readers, it clicked. Here was a system that felt modern. It used the language of blood, genes, and types, but functioned socially like astrology or the classical four humors. It gave people an easy script. Type A, serious, organized, considerate, maybe a bit anxious. Type B, individualistic, passionate, unpredictable. Type O, confident, decisive, sometimes stubborn. Type A, B, mysterious, hard to read, a mix of opposites. The match was rarely perfect, but it didn't need to be. Television variety shows ran segments on blood type compatibility. Magazines published charts. Dating services and social clubs used it as an icebreaker. In South Korea, similar ideas spread later, sometimes framed as light-hearted fun, sometimes taken more seriously. Over time, entire microcultures formed around these four letters. People shared stories of relationships that proved the compatibility charts. Parents compared their children's types and nodded knowingly at their differences. Office workers joked that the new hire acts just like a bee. From the outside, especially from a scientific perspective, this can look puzzling. Study after study finds no robust link between type and personality traits when you measure them carefully with standardized tests. Whatever faint patterns appear tend to vanish or flip when the sample changes or the method improves. Exactly what you would expect if you're mostly measuring noise, but from the inside, the blood type stories feel convincing. There are good reasons for that. One is confirmation bias. When you hear that type A people are conscientious, you don't test that claim against every type A person you know. You notice the colleagues who fit the description, the friend who always arrives 5 minutes early, the cousin who color codes their planner, and you say, that's so A. The people who don't fit, they slip out of focus. The same happens for type Bs who are messy, Os who like being in charge, Abs who seem contradictory. The matches stick in memory, the mismatches dissolve. Another is social reinforcement. If your peers talk about blood types, you pick up the language. At parties, in classrooms, online, people ask, "What's your type?" If you say you're B, and everyone laughs and says, "Oh, that explains it," you get a small, immediate reward. You feel seen, included. Over time, you may even exaggerate or downplay certain traits to match what others expect of your type. Personality isn't fixed. It's shaped by feedback. A folklore system can become a gentle script that people act into. Then there's the comfort of identity. Being human is confusing. We're bundles of contradictions, changing across situations and years. Any tidy label that promises to distill that complexity into a few adjectives is tempting. It offers a story about who you are and why you are that way. A story that does not blame your choices or your upbringing, but something neutral and shared. My blood. It can also make other people's quirks more tolerable. He's not being difficult. He's just an O can soften frustration. The way she's a Leo or he's the oldest child sometimes does. Blood type folklore, then, is not primarily about biology. It is about social needs. When you zoom back out to the actual immunology, the contrast is stark. Antigens are specific sugar molecules attached to the surface of your red blood cells and some other tissues. They are built by enzymes encoded by the gene which either add certain sugar units to a base, structure for A and B, or leave it unmodified for O. Your immune system learns to ignore the antigens your own cells carry, and to treat the others as foreign. That is why a type A person has anti-B antibodies, type B has anti-A, type O has both, and type AB has neither. Those antibodies are small Y-shaped proteins that circulate in your plasma, ready to bind to mismatched red cells and flag them for destruction. This is crucial for defense against pathogens, but it can be catastrophic if incompatible blood is transfused. That's the core of biology. Chemistry on the cell surface, proteins in the blood, and the finely tuned pattern recognition system we call the immune response. Nothing in that picture points naturally to introversion or leadership style. The evolutionary pressures that shaped variation may be cholera in one region, malaria in another, other pathogens and clotting risks elsewhere, operated on survival and reproduction, not on whether you like small talk. The stories that say otherwise are part of a different layer of reality. They are cultural adaptations, not genetic ones. They spread because they are catchy, not because they are true. They help people navigate relationships and social expectations, at least in the short term. In that sense, they have their own kind of fitness, but they also carry ethical risks. When a light-hearted typology becomes a serious basis for judgment, it can slide into stereotyping. In Japan and South Korea, there have been documented cases of what the media call blood type harassment. People being teased, excluded, or even disadvantaged at work or school because their type is associated with negative traits. Employers have reportedly asked job candidates for their blood type. Children have been bullied with it as an excuse. From a medical standpoint, misinformation can be more quietly harmful. If someone hears that a certain blood type is strong against disease, they might ignore screening or downplay symptoms. If another type is described as weak, people may worry unnecessarily about conditions that have little to do with folk claims about diet or exercise based on blood type can distract from more solid advice about nutrition, sleep, and activity that applies across all groups. So, how do you enjoy the cultural side of blood types without getting pulled into pseudoscience? One approach is to treat these narratives as what they really are, modern folklore. You can appreciate them the way you might appreciate horoscopes, personality quizzes, or traditional stories about apicious birth years. They can be conversation starters, bits of shared language, small windows into how different societies mix old ideas with new science. You can smile when someone says, "That's such a thing to do," without assuming that their blood cells actually dictated their behavior. At the same time, you can quietly keep two categories in mind. In one category sits the measurable biology of antigens and antibodies, the evolutionary history of pathogens and populations, the clinical realities of transfusion reactions and Rh-incompatible pregnancies. This is the domain where blood type truly matters for your body. In the other sit the stories we build around those facts. Stories of personality, compatibility, destiny. Some are playful, some are earnest. All of them tell you far more about human imagination than about glycans on red blood cells. The real mystery of blood types, the one that science keeps chipping away at, is not mystical. It lives in evolution and history. Why did these particular antigens arise when they did? How did ancient epidemics and migrations sculpt their frequencies across continents? Why do some rare types cluster in specific populations? And what does that tell us about old bottlenecks, founder effects, and vanished ways of life? Those are questions that connect your veins to Neanderthals, Denisovan, medieval plagues, tropical parasites, and the logistics of 20th century wars. They are strange and beautiful enough that we do not need to give supernatural powers. We can let science handle the part of the story written in sugars and proteins, and let culture handle the jokes, the myths, and the small comforts of saying, "I'm A" or "I'm O," and seeing someone else nod in recognition. In the next section, we'll leave the realm of folklore and return to that deeper puzzle. We'll look at what we still don't know about blood types, where current research is heading, and how many questions remain hidden in something as ordinary as a drop of red. For all the detail we've gone through in this series, there's something humbling we need to sit with. When it comes to blood types, especially, and Rh, we're still very much in the middle of the story. We have good outlines, strong hints, some very convincing pieces of the puzzle. But the full picture, the exact reasons these variants exist in the combinations and frequencies we see today, that picture is still coming into focus. Right now, most researchers think about and Rh diversity as the result of several forces acting together rather than one grand cause. Natural selection is one of them. Different blood group variants seem to offer advantages or disadvantages depending on the diseases, diets, and environments people face. Type O can reduce the risk of severe malaria in some contexts, but it may make cholera infections more intense. Type A may have been favored in regions where certain pathogens stuck more easily to type O cells, or which negative might have been tolerated or even slightly favored in places or times when particular infections or pregnancy patterns made its risks less severe. But selection is only part of the story. Genetic drift also matters. That's the random side of evolution, the way of frequencies can change just by chance when populations are small. Imagine a hunting party of 20 people, or a village of a few hundred, where just a handful of births and deaths can shift the local balance of A, B, O, and R. Over thousands of years, some groups might lose a blood type variant entirely, not because it was harmful, but simply because no one carrying it happened to leave descendants. Migration adds another layer. As people moved, they didn't carry the global mix of blood types with them. They carried whatever mix existed in their particular band or community. A small clan with mostly type O leaving Africa might seed rich populations in distant lands. Traders or pastoralists from a region with more beleys might carry that variant along steppe, routes, or river corridors. Over time, waves of movement, intermarriage, conquest, and isolation stitch together a patchwork of blood type distributions that still echo ancient journeys. And then there are trade-offs. The same variant can be helpful in one context, harmful in another, and neutral in a third. Type O's apparent malaria advantage may have kept it common in some tropical regions, even if it raised the risk of bleeding or certain infections. Type A or AB might slightly increase the risk of clots or particular cancers, but reduce susceptibility to some bacteria or influence fertility in subtle ways, which negative dangerous in some pregnancy combinations might interact with other genes to change immune responses in ways we don't fully understand yet. Selection, drift, migration, trade-offs, four forces weaving together, not in a tidy pattern, but in overlapping, sometimes conflicting threads. The open questions mostly live in the details. We still don't know which pathogens mattered most, at which times, and in which places for each blood group system. Was cholera really a major sculptor of frequencies along river deltas and coastal plains? Or are we overreading a pattern from the last few centuries? How much did smallpox shape the prevalence of type A in Europe and parts of Asia? And did that pressure operate over millennia, or mainly in the last 1,000 years? Did different malaria species in Africa, India, and Southeast Asia tug and Rh frequencies in slightly different directions? How important were gut parasites, respiratory viruses, or unknown bacterial infections that no longer circulate today, even for Rh, which seems so straightforward when we focus on pregnancy? The deeper story is murky. Were there specific infections where B and OH positive was a liability, nudging Rh negative to persist in certain populations despite its costs? Did patterns of family, structure, birth spacing, and fertility in different cultures change how strong Rh selection really was? The archaeological and written records show shifts in childbirth practices, breastfeeding duration, and maternal mortality over time. Each of those could subtly change how Rh incompatibility played out in real communities. We also don't fully understand the timing. Did most of the shaping happen far back in the Pliocene, when small scattered human groups were dealing with local parasites and harsh climates? Or did later urbanization, agriculture, and crowd diseases, plagues that needed dense populations to thrive, reshuffle blood type advantages in the last 10,000 years? To move from broad stories to precise answers, researchers are leaning heavily on new tools. Genomics lets us scan entire genomes from hundreds of thousands of people and ask, where has natural selection left a fingerprint? Regions around the Rh genes show signs of what geneticists call balancing selection. Patterns suggesting that multiple variants have been maintained over long periods, instead of one winning and sweeping to fixation. By comparing these signatures across continents, we can begin to infer when selection was local and when it was global. Large biobanks add another dimension. In places like the UK, Finland, Japan, and increasingly in other countries, volunteers have given blood or saliva samples along with detailed health records that allows researchers to link specific blood types and even finer variations within the Rh systems to hundreds of medical traits, clotting disorders, infection outcomes, pregnancy complications, autoimmune diseases. Most of the effects are modest, but taken together, they help map out the trade-off landscape. They show, for example, how type O's lower clotting factor levels might interact with modern cardiovascular risks, or how certain rare R variants behave in pregnancy. They also highlight associations we didn't expect, which can then send lab researchers back to the bench to look at how particular pathogens stick or fail to stick to different cell surfaces. Ancient DNA is pushing even deeper. Early studies could only tell us rough ancestry components. This skeleton looks more like modern Europeans, that one more like East Asians or indigenous Americans. Newer work is precise enough to call some blood group alleles directly from ancient bones and predate, as more Neanderthal and Denisovan genomes are sequenced, and as we get higher quality DNA from early farmers, hunter-gatherers, and pastoralists, we'll be able to trace and Rh variants along specific migration routes. We'll see when a particular allele first appears in a region, when its frequency jumps or drops, and whether those changes line up with evidence of new diseases, climate shifts, or population turnovers in the archaeological record. Piece by piece, timelines will sharpen, mechanisms will move from maybe to probably, from broad speculation to well-tested models, but even with perfect data, there's a deeper reason the story will never reduce to a single decisive cause. Complex traits in human evolution almost never have one explanation. Height, skin color, lactose tolerance, mental health vulnerabilities. Each turns out to involve many genes, many environments, and many historical contingencies layered together. Blood type diversity is no different. There was no single ancient epidemic that created type B. No one river valley that invented Rh negative. Instead, there were countless small events, births and deaths, migrations and isolations, fevers survived and pregnancies lost, trade routes opening and closing, climates warming and cooling. Each event nudged frequencies a fraction of a percent. Over tens of thousands of years, those nudges accumulated into the patterns we see now. Holding that kind of complexity in mind can feel unsatisfying at first. We often want a clean narrative. Malaria did it, or plague did it. A single villain or hero that explains everything. But the real story is richer, and in a way, more human. It reflects the messy, branching, uncertain paths our ancestors actually walked. As we explore that uncertainty, this is also a good moment to mention something new we've been working on. It goes into greater depth on how scientists piece together these invisible histories, and how much we still don't know. For now, the most honest place to land is somewhere between confidence and mystery. We can say with confidence that blood types matter for transfusion, for pregnancy, and for certain infections and clotting risks. We can say that their global patterns bear the fingerprints of selection, drift, migration, and trade-offs operating over immense spans of time. At the same time, we can admit that many specifics remain fuzzy. Which outbreaks in which centuries tipped the balance toward or away from a given AL? How interactions between blood groups and other genes shaped fertility in particular cultures? How many ancient pathogens have disappeared without leaving a clear trace, taking their side of the story with them? Learning to sit with that is its own kind of skill. To look at a drop of blood and know that part of its history is clear, mapped in textbooks and hospital protocols, while another part fades into mist somewhere between Neanderthals, Denisovans, and unrecorded epidemics. In the final section, we'll gather these threads together. We'll return to the idea we started with, that there is an ancient mystery still inside us, written, not in prophecy or personality charts, but in the quiet, persistent diversity of the blood that keeps us alive. So now we're back where we began. Somewhere inside you, circulating quietly while you listen to this, are a few small molecular differences that we call a blood type. A, B, AB, or OH positive, or RH negative. Just a handful of sugars on the surface of your red blood cells, and a protein that may or may not be there. On the surface, it's chemistry, straightforward biochemistry. Enzymes that add or don't add a tiny sugar. Antibodies that recognize self and not self. Lab tests that sort blood into neat columns and cross-matching tables that tell doctors who can safely donate to whom. But once you step back, those same little sugars stop looking ordinary. They become the fingerprints of history. They carry the legacy of trial and error medicine when physicians tried transfusions without knowing why some patients survived and others died. They carry the story of a young scientist in the early 1900s watching red cells clump under a microscope and realizing that humans were not all the same inside. They carry the slow building of compatibility charts, blood banks, Rh testing in pregnancy, and the quiet routines that now save lives every day in hospitals around the world. They also carry something older. Every time your heart beats, it's pushing through your arteries. A record of ancient battles you never saw. Malaria parasites learning how to invade red cells and your ancestors' cells changing in man response. Bacteria in river water and coastal estuaries binding more easily to one blood group than another. Viruses riding through crowded cities, taking a slightly higher toll on some types and sparing others. Plagues we know by name, Smallpox, Cholera, and others that vanished before anyone wrote them down. Over and over, tiny differences in risk, a few percentage points here and there, added up. Someone with one blood type survived a fever long enough to have children. Someone with another type had fewer pregnancy complications or fewer deadly clots in one particular environment. In a different place or time, the advantage flipped. What was helpful in a malarial swamp could be harmful in a freezing steppe or in a crowded medieval town. Layered on top of that is movement. Bands of hunter-gatherers carrying their local blend of and Rh as they followed herds and rivers. Early farmers and herders spreading out from a fertile valley, bringing not just crops and animals, but their particular genetic mix. Traders crossing deserts and seas, marrying into distant communities and stitching together far-flung bloodlines. Each journey slightly reshaped the map of blood types, leaving traces that can still be seen in global patterns today. Underneath all of it lies deeper time, the fused chromosome that marks us as descendants of a small ancient population. The echoes of Neanderthal and Denisovan DNA, including blood group variants they carried and passed on when our lineages met and interbred. The long shadow of prehistoric epidemics, climate swings, and migrations that no one recorded. But that left subtle signatures in the genes that control the antigens on your red cells. So when you hear type A or type O on a hospital bracelet, you're hearing a label that sits at the intersection of molecules, medicine, evolution, disease, and human movement. It's a practical label. It matters if you ever need a transfusion, or if you're pregnant and Rh negative. It nudges your risk of certain infections and clots, very slightly up or down. For most people, most of the time, it quietly does its work, without drawing attention to itself. But it's also a story that began long before anyone wrote or farmed or built cities, a story of survival in small, scattered groups, of bodies adapting, not perfectly, but well enough to a changing, dangerous world, of different human populations meeting, mixing, and moving on, of ancient relatives like Neanderthals and Denisovans, leaving faint but real traces in the way some of our blood groups are structured today. The main thread running through all of this is that your blood type isn't a personality category or a mystical sign. It's living history. History shaped by pressures we can partly trace. Malaria here, Smallpox there, cholera along certain coasts, and by many more forces we can only glimpse. As our tools improve, that story will get sharper. More genomes sequenced from living people. More ancient DNA pulled from teeth and bones. Better models of how pathogens interact with specific blood group variants. Richer medical datasets linking subtle health differences to particular alleles at the and, rosy, and beyond them, to other blood group systems that rarely make headlines. But even then, some pieces will probably stay in shadow. We'll never know the names of the first people who carried the AI, or the first small group whose high frequency of O happened to survive a local epidemic. We may never reconstruct every vanished pathogen that nudged arch frequencies up or down in a particular valley 10,000 years ago. Some parts of this history will remain like distant stars. We see their effects, but not their full shapes. That uncertainty can be a place to rest, not just a gap to be filled. To fall asleep perhaps, to the idea that every breath you take is oxygen, moving through a system, refined by countless ancestors, facing challenges you will never meet. That the same blood type you record once on a medical form is also a quiet message from people who lived and died tens of thousands of years ago. Before we close, if you've enjoyed drifting through this mix of genetics, archaeology, and deep time, it would really help if you tapped like on this episode, left a comment, and subscribed so you can find your way back here next time. If you feel like sharing, I'd love to know where you're listening from. What city, region, or landscape is wrapped around you while you hear this story about the landscapes inside your veins? For now, you can let the details fade if you like, and just hold on to this. Inside you right now, there is an ancient mystery still at work. Not a riddle to be solved in one neat answer, but a long branching history written in the small differences that keep your blood flowing safely through your body. A history that ties you to epidemics and climates, to forgotten rivers and long-vanished relatives, to all the people who made it through long enough for you to be here, listening, with that same quietly remarkable blood moving through your veins.